to thank you for allowing us to, again, have Dr. Lytton share his life, his legacy with us today. So again, we'll move on to the next part. And
I've lost a good friend.
He was a gentleman. He was always kind.
The things when you think about Dr. Lytton were his, boundless energy, energy which would put a resident to shame, even well into his 60s, and his surgical fearlessness, just an absolutely fearless surgeon, afraid of nothing. But the word I thought that best characterized him was grace, just a guy who watched out for everything and everyone, kind, thoughtful, and just a great leader.
He was friendly, he was approachable. I never saw him dress anybody down in a nasty or negative way. We did a lot of fun things together. We skied together. We did some mountain climbing together. He was a very physically active guy who had a great zest for life.
He encouraged the staff and the residents to think creatively, to work hard, to be honest, to have integrity, to be motivated by what's best for patients. And for that, he led by example. He was an amazing physician, surgeon, department chair, educator, and leader.
He carried himself with an elegant authority, but he was always gracious with his time, his teaching, and his advice.
The first thing that comes to mind was that he was the true Renaissance man. Not only was he a tremendous surgeon and teacher, but he was so well versed in so many fields. He loved the arts, he was well read, and he could discuss virtually anything with you.
You meet very few people like him from the onset. He was tall, he was stately, he was proper, he was friendly. He was the type of person you meet once or twice in your lifetime. Well, first of all, he could do anything surgically. He was fearless.
After a surgical career of my own of over 50 years, I think that he's the best surgeon I ever operated with. I was fortunate to be with him when we did the first kidney transplant at Yale, and he was 45 at the time, and he sold his competence to the powers that be, and I'm not sure what he did to hone his skills, because he hadn't been doing that sort of surgery on a regular basis, but he did it and made it look like he'd been doing it every day.
He was influential in, one, setting up with the nephrologist the first dialysis center in Connecticut. He brought various programs, not inventing them, but programs that people had done in other places and were just in their infancy, such as percutaneous nephrolithotomy. A lot of the bowel work, which is done today for bladder replacement surgery, Bernie was one of the early people in fostering it.
I especially remember the first time I did a ureterochalacostomy and had him assisting me, which gave me the confidence I needed to do that difficult operation.
He was so meticulous as a surgeon, and he truly was one of the finest surgeons I think I've ever seen.
We went together, flew to Detroit and watched one of the original persons who started robotic surgery at the time. So he was always interested, even though he had retired or stopped practicing. He was always very inquisitive, very wanting to know what things were going on in urology.
I came out to Mulberry Point in Guilford, which was the site of so many of Dr. Lytton's famous resident summer picnics, and it just seemed like a fitting place to pay tribute because of the fond memories.
They respected him. They considered him a friend. I don't think any of them would have gone in there and say, "Hi, Dr. Lytton." He'd be, "Hey, Bernie, I got this problem."
As the first female in the program, visiting professors of a certain ilk, might I say, were quite inappropriate. He took one of them aside privately, chastised him, and withheld his honorarium. In my experience in the early 1980s, this was singular in the support given trainees. He had our backs always.
One of the nurses came to me and said, "Dr. Krause, Dr. Lytton's on the phone, and he doesn't sound very happy." So I picked up the phone, and it was Dr. Lytton. He said, "Krause, where are you?" And I said, "Dr. Lytton," I said, I'm stuck. I'm stuck making rounds. I said, every room we walk into is a disaster, and they're all your patients." Well, he said, "Okay, do what you got to do and come up when you can." You know, and that's just the kind of guy he was. You know, I mean, he didn't pull rank, not ever.
And he worked with an economy of motion that we all tried to imitate. Whenever we had a difficult clinical patient, not only would we ask ourselves, what would Dr. Lytton do? But we'd also ask, how would Dr. Lytton do it? And that always seemed to be the right answer.
He was pioneering continent diversions while I was at Yale, and I can still hear his voice when I do those cases now saying, "Oh, take a little bit of mucosa and a lot of serosa, a little bit of mucosa, a lot of serosa." His voice still rings in my ears even now, almost 40 years later after I trained with him.
The last thing he wanted to do was disappoint Dr. Lytton. And if he did, it didn't happen again. He just did it once. And he didn't have to tell you, he didn't have to raise his voice at you, he didn't have to reprimand you. You just knew if you did something wrong. And it just didn't happen again.
And then we had a case to do, and it was his case, and I was to help him. And it started off the case with him asking me for instructions about a whole lot of things. But by the end of the case, he was saying stuff like, "You know, Krause, if you were a better cameraman, I'd be able to get this done a lot quicker." That was, yeah. I guess he was funny.
With a twinkle in his eyes, he made a gentle joke, with a broad, infectious smile when he was amused by a comment or a situation. I will always remember that smile, warm, genuine, never used to chide or with cynicism.
But Dr. Lytton could also at times be a bit of the absent-minded professor, and that was part of his beauty. I fondly remember the time, and this was well before cell phones, when I was at home in my apartment just relaxing one evening, and he calls me up at home and says, Fred, where are you? Where are you?" I said, "Dr. Lytton, I'm at home. You called me here." Oh yeah, well, come down and do a case with me."
Did you pull any pranks on Dr. Lytton?
None that I wish to tell you.
In one of my first surgeries with him, deep in a bloody and well-adhesed pelvis, I found a structure he had been searching for 5 minutes before he did. His response? "Well, Vito, even a blind squirrel comes across a nut now and again."
Probably, other than my father, the most influential male person in my life.
But I think probably the most important thing he taught me was that you could lead a division and be personal and not have to sit on a throne.
I kept in very close touch with him during my whole career. I feel exquisitely indebted to him for what I did and what I became. And I think that you will hear that sentiment echoed by anybody who was in my position.
I cannot imagine having a better urologic training than I had under his direction, nor a better personal relationship with the program director than I had with him. He leaves behind a tremendous legacy.
I wish you were here to hear these words. I can't thank you enough for everything that you ever did for the Yale University Urology Department, its residency program, and for my career.
I also want to thank his family. Thank you for sharing him with us. That was a special gift that has a lasting impact that will always be remembered.
In the military, it is said that you die twice, the first time when you draw your last breath, and the second when the last person on earth says your name for the last time. Today, we show that the world will continue to say the name of Bernard Lytton for many, many years to come.
God bless him, and may his memory be a blessing for us all.
On October 24th, 2022, Bernard Lytton, a respected innovator, clinician, and educator, and my mentor, friend, and colleague, died in England at the age of 96. Bernard was born in England on June 28, 1926, attended Haberdasher School, and obtained his medical university at the University of London, England, in addition to obtaining his surgical and urologic training at the London Hospital. He trained in Johannesburg General Hospital in South Africa and the Royal Victoria Hospital, McGill University in Montreal, Canada. In the midst of his training, he spent 2 years during the Korean War in the Royal Air Force, attaining the rank of Squadron Leader Major. In the early 1960s, at that time, the United States had a more distinct specialty of urology than was seen in the UK, and Bernard, in 1962, emigrated to the United States and was recruited to Yale by Clyde Deming and B. Marvin Harvard. In 1967, he became chief of urology, a position he held for 20 years. Bernard was a mentor, a benevolent leader, a team builder, and a gifted surgeon. He rapidly recruited 3 young urologists, Marty Schiff, Ed McGuire, and myself, and we had fun. We were all friends, and within 10 years, we were all Yale full professors. In those days, it was up or out. In the 1960s, in partnership with Howard Levitin and the section of nephrology, he opened the first dialysis unit in Connecticut, and he performed the first renal transplant in Connecticut in December 1967. He instituted programs to provide brachytherapy for prostate cancer, percutaneous nephrolithotomy, continent urinary reservoirs, and bladder substitutions. He also transferred what he had seen in the UK and was instrumental in setting up the first hospice facility in the United States. Lytton, in his early years, made basic scientific contributions, addressing the etiology of compensatory renal hypertrophy and cancer immunology. In London, he had taken a seminar with Rupert Billingham, Leslie Brent, and Peter Medawar on their classic work on immunological tolerance, for which Peter Medawar received the Nobel Prize. That seminar sparked Lytton's career-long interest in immunology. He served on the surgery, anesthesia, and trauma study section of the NIH from 1975 to 1979. Bernard, also known as Lytton from Britain, the Lord of the Flies, was an excellent teacher and received the Francis Gilman Blake Award, which is given by the senior class at the Yale School of Medicine to the most outstanding teacher of the medical sciences. This was most unusual for a physician, for a urologist, that is, because it usually went to primary care physicians. In fact, Bernard is the only urologist to have received this award. Here we see him receiving the Hugh Hampton Young Award from the American Urological Association. Bernard was an honorary member of the American Association of Genitourinary Surgeons and served as president of the New England section of the AUA, the Clinical Society of Genitourinary Surgeons, and the American Association of Genitourinary Surgeons. Bernard avidly played tennis, played squash with his residents, hiked in Nepal, and enjoyed skiing, sailing, and his home on the Connecticut shore. Bernard loved family, and just as he was persistent and resilient during tough surgical cases, he never let setbacks destroy him. In his later years, he bounced back from a severe head injury. Furthermore, he was married and had 2 children, Sharon and Simon, before coming to the States. While packing his household to move to the States, his wife, Helene, was taken ill with influenza and died. He subsequently married Norma Lytton, had a wonderful life with 2 more wonderful children, Tim and Jenny, only to lose Norma to breast cancer. He spent his final 11 years both in the UK and in the United States with his loving partner, Dawn Wood. In his later years, Bernard was appointed to be the president, by the president of Yale, to important positions in the university, and was the master of Jonathan Edwards College and the founding director of the Kerner Center for Emeritus Faculty at Yale. When I was in Greenland in the service 800 miles from the North Pole, I learned the first law of the Arctic. Only the lead dog has a changing and interesting view. To the rest, the view is rather monotonous. Bernard Lytton was a leader. Most importantly, Bernard was my friend and colleague. He will be missed. Thank you.
Okay, again, thank you, Dr. Weiss, for that beautiful eulogy. So then we'll move on to now the rest of the symposium, and it'll start kick it off with Dr. John Libertino. I want to just, again, he needs another speaker this morning who needs no introduction, but just to give you, for our trainees, we're here. So again, Dr. Libertino graduated from Georgetown University School of Medicine, and he's an alumni, finished his training back in 1970. He's currently a professor of urology at Tufts, and he has held various leadership positions, including CEO, chair of surgery, vice chair, etc., at Leahy Clinic. He's also been a frequent contributor to journals, has been editor-in-chief of the American Journal of Urology Review. Again, Dr. Libertino, thank you for coming this morning. And I think he's going to be giving us a talk about the surgical techniques, results, and new paradigms for the management of high-risk T3 and T4 renal cell carcinoma. Thank you, Dr. Libertino.
So what I'd like to do is talk about what I've been involved in for many years, and that is the current surgical techniques and their results with a potentially new paradigm for the management of high-risk T3 and T4 renal cell cancer involving the vena cava. Now, this is the reflection of this team of physicians that I worked with. Dr. D'Agostino was the cardiothoracic surgeon, and Dr. Piemonte, the interventional cardiologist. And you will see that this kind of work needs to be done by a team. It's not an individual effort. So what I'd like to do today is to talk a little bit about the evolution of where we are today and how we got here, and then talk about a new paradigm for the future. During my surgical career, I've had the opportunity of treating well over 347 patients who underwent radical nephrectomy with venous extension. 182 of those patients had the diagnosis of T3B or T3C renal cell cancer. We analyzed this group by two methods, the AJCC and the Mayo thrombus classification, to see if one system was better in determining prognosis than the other. And we looked at 21 potential risk factors, and in addition, looked at rates of complication, tumor recurrence, intraoperative death, and 30-day mortality. Now, under the AJCC classification, when we stratified our experience, we had 130 with T3B, which was tumor up to the diaphragm, and 60 patients who had aortocaval and atrial tumor extension. We analyzed this and published the risk factors that affected survival in patients with renal cell carcinoma invading the cava in 2017. And of those 182 patients analyzed, there were several things that stood out as being biopredictive. They were the clear cell histology, positive lymph node involvement, and perinephric fat involvement. And if you look at the pathologic stage, T3, B, and C, you will see that the level of the tumor thrombus was not statistically significantly important in determining prognosis for these patients. Of the 182 patients, the overall survival at 5 years was 32%. The disease-specific survival was 40%. And as you can see, the tumor-free survival tracked pretty much the disease-specific survival curve. So we learned one lesson from this analysis, and that is that in T3B and T3C renal cell cancers, the overall survival was impacted and associated with lymph node positivity, perinephric fat involvement, clear cell histology, but did not and was not really statistically significantly affected by the level of the tumor thrombus. So that ended the shibboleth that was being debated in the literature for about 20 years. We also then stratified the same group of patients according to the Mayo Clinic categorization. And you can see here level 3 are tumors that are extending above the hepatic vein to the diaphragm, and level 4 tumor is intracardiac disease. So we had an early interest in, and everybody talks about being fearless, and both my chairs were fearless. Everybody knows that Dr. Lytton was a fearless surgeon. So when it came time to be fearless, I think we stepped up to the plate and developed the use of total circulatory arrest for the management of cavoatrial tumors. Now, this operation we published in, I guess, 1990. And you can see here is the tumor thrombus extending up into the lollipop extension of the tumor into the atrium. And these patients were treated with the so-called Mercedes-Benz incision, which was a median sternotomy and a chevron incision. Initially, in the first 23 patients, what we did was we mobilized the tumor and the entire vena cava before putting the patient on bypass, because we were afraid and concerned that putting patients on bypass, you would anticoagulate them and they would bleed more. But in the 23rd patient, utilizing this approach, we had a patient who threw off a piece of tumor thrombus, had a patent foramen ovale, went up to the carotid, and had a stroke. So we thought it was time to change the approach. So we did do that, and in 1998, we reported the use of the minimally invasive access approach for cardiopulmonary bypass. And in this technique, what we did was use a chevron incision, and we put the patient on bypass initially and anticoagulated them, and avoided the use of a median sternotomy by using the subclavian artery for arterial return. And in this approach, the cardiac surgeon would put the patient on bypass, open the atrium, right atrium, take out the tumor thrombus, and we would open the vena cava below the hepatic veins, take out the tumor thrombus, and then reconstruct the vena cava. At the time that was accomplished, the cardiac surgeon was closing the heart, we closed the vena cava, and then we did the radical nephrectomy and mobilization of the kidney after the vena cava was closed and patient was off bypass. We then looked at those 70 patients and compared the median sternotomy group with the minimally invasive group, and there were 23 in the median sternotomy and 47 in the minimal access approach. And the overall survival was better with the minimally invasive approach, and it makes sense when you see that you obviate a lot of the complications that were occurring using the median sternotomy. As we studied these patients, it was very interesting that the recurrence-free survival was also better with the minimally invasive approach, and people couldn't understand why that could be. But I think what was happening is patients who were free of disease were dying of the complications of cardiopulmonary bypass. And so therefore, when we looked at the minimally invasive approach, they had less surgical complications and better recurrence-free survival. So the second observation is that with this approach, we had a shorter operative time, shorter length of stay, less days of intubation, less wound infection, less pneumonia, less ATN, far less use of inotropic medications. And interestingly enough, we had no evidence of any tumor thrombus recurrence in either of these groups. And I think that was because we were using cardiac arrest. We had a stable surgical field, could take out the tumor completely without being rushed, being concerned about blood loss. In addition, I think there was a major decrease in hospital mortality and a longer median survival using the minimally invasive approach. And so the second lesson we learned was that this approach was better and significantly better than the traditional median sternotomy. And so that's how we evolved to getting to this point of using the minimally invasive approach for patients with level 3 and level 4 renal cell cancer. Now, the critical junction for using cardiopulmonary bypass is if the disease is above the hepatic veins, because we've done a significant number of patients with this level of tumor thrombus, and you can do these off pump, and they're difficult, and I'll show you what we did. But the critical junction is anything above the hepatic veins or into the hepatic veins really does require cardiopulmonary bypass. And so we looked at our level 3 and level 4 experience using the Mayo Clinic staging system. And in that group, we had 93 patients, 70 who we treated with cardiopulmonary bypass and circulatory arrest, and 19 who were treated with a Pringle maneuver without bypass. And that experience was pretty satisfactory, but then We had a gentleman referred up, interestingly enough, from Connecticut who had presented with a large pulmonary embolus, which precluded cardiopulmonary bypass. My colleague, Dr. D'Agostino, said we cannot put him on bypass, we'll kill him because of the cytokine storm that will occur after we take him off bypass. And so, although we worked him up completely, you can see that the arrows are pointing to the tumor, which is not large, and to the caval thrombus, which was thin and delicate. And you can see on the second frame, the intrahepatic portion of the thrombus. But I think Jody, I just screwed this up. Let me see if I can fix it up. Yeah, I think that's, I found it. Thanks. I was trying to play a video, and this is his cardiac echo, and you can see the tumor thrombus in the ventricle abutting the pulmonary valve. And so we were not able to do or even consider doing a circ arrest and extraction of this tumor. And he was 53 years of age, and he had 3 children and a dedicated wife who begged me one night to think about a different approach. And so I did. I went and begged our interventional cardiologist, Tom Piemonte, to do this endovascularly. There was very little precedent for this. And certainly in this case, necessity was the mother of invention. So what we did was we put the patient on ECMO, and we advanced the angiovac, which was a device that one of the cardiac surgeons and I developed to take tumor thrombi out of the pulmonary artery. But instead, we passed it down into the vena cava and evacuated the tumor thrombus using the angiovac device. And you can see here, this is the tumor thrombus that was extracted through the IJ vein. And what was left was a very small component of the tumor at the level of the renal vein. This was simply taken care of by doing a partial caval resection distal to the thrombus and reconstructing the vena cava with 3-O-proline. We reported this, and I believe, and I know it was the first reported case of cardiac endovascular extraction of a tumor thrombus. And that was done in 2016. We then had several other patients. This is a 74-year-old gentleman who presented with a PE, had the same issues with cardiopulmonary bypass. And so what we did after his evaluation showed that he had a right atrial tumor extension. We angioinfarcted the right kidney, and we proceeded with what I dubbed endovascular downstaging. We wanted to get this tumor down below the hepatic veins, and you can see in the canister the fragments of the tumor that were taken out with the angiovac. We then were able to get Rommel tourniquets below the hepatic veins and below the tumor thrombus in both renal veins and extract that tumor thrombus without bypass. And you can see the right plate showing the reconstruction of the vena cava using 3-O-proline. We also had another patient, similar circumstances, with tumor extending, as you can see, into the level of the hepatic veins. And you can see in the middle panel, the tumor that was removed. And you can see, if I can get the arrow there, what the cavogram showed after the tumor thrombus was removed. And that was very straightforward radical nephrectomy with a small resection of the vena cava. So in conclusion, here's where we are today. Neither staging system defines prognosis. What the tumor level is not biopredictive of outcome. It is the biology of the tumor that's much more predictive. The minimally invasive cardiopulmonary bypass approach is better than the median sternotomy. It reduces morbidity and mortality, therefore reducing surgical risk. And I believe that endovascular downstaging also reduces surgical risk because it obviates cardiopulmonary bypass. And it is something that can be done after this is done by your interventional cardiologist, by most urologists, even in a community hospital setting. So the new concept that I'd like to present is that of endovascular downstaging to reduce the tumor thrombus below the level of the hepatic veins. It makes the procedure technically less difficult. It obviates all the problems of bypass. It reduces morbidity and mortality, and therefore surgical risk. And quite frankly, the lower the tumor thrombus, the better it is for the patient, because certainly avoiding cardiopulmonary bypass complications is an advantage. It provides shorter OR time, less blood loss, and shorter length of stay. So all of these things reduce surgical complications and reduce surgical risk. So I think there may be a future for this paradigm. I hope you'll think it through with me. But I do think it's worthy of your consideration because quite frankly, anything you can do to make this operation simpler and less complicated, I believe it makes it safer and more effective. So I can't have thanked Dr. Lytton enough for selecting me as his first resident, bringing me to Yale, which I came to love. My experience here was transformative. He shaped me in many ways. He opened my mind to the past by introducing me to the rare books library at the medical school, which I loved and spent most of my time on call reading ancient history. But it did provide me with the foundation of thinking about how to make things better. And it also made me understand in a better way how we got to where we are today and how together we can explore future paradigms together. And as I said in that video, I truly wish that Dr. Lytton was here today to hear our celebration of his life, to learn and so that he would hear of our appreciation, admiration, and respect that we had for him. And that means everybody in this room. So thank you so much for inviting me. I don't know about questions and answers because Jody didn't help me.
Let's say questions if there's any from the audience.
Yes, Paul. Angiovac. It's I believe it's commercially available. I did this with one of the cardiac surgeons at Leahy and developed this, and then he went out to Phoenix, and this device became very successful for him from a business perspective, and I believe he gave up cardiac surgery as a result of this development. Thank you.
Thank you, Dr. Libertino. Okay. Thank you. So now we'll move on to the next speaker, Dr. Michael Leapman, who is our junior faculty member. Can I say junior or no? Yeah. Junior faculty. It's really, again, I think one of the objectives of this meeting was to really introduce some of our newer faculty members that some of you may not be familiar with. But Dr. Leapman has been doing exciting work since his arrival in our department. Dr. Leapman graduated from University of Maryland. That's where he did his medical degree, MD, and finishes his residency training at Mount Sinai Medical Center in 2014. Completed his fellowship at the University of California, San Francisco with Dr. Peter Carroll. And he's currently our faculty member, associate professor in our department. He also has a secondary appointment at the School of Public Health and also chronic disease of epidemiology. He also leads our prostate and cancer programs. He'll be speaking to us this morning. He's got many research going on, but the topic that he'll be speaking this morning is development and outcome of a longitudinal prostate cancer transcriptomic in real-world clinical data usage. Or that's linkage.
Thank you so much. So thank you so much for the honor and opportunity to speak here. I want to get one disclosure out of the way. I am going to talk about a research collaboration that's uncompensated with Veracyte, who's the maker of Decipher, which is a genomics platform. So the outline for this morning, I want to begin with a comment on gratitude and discovery, I think in honor of Dr. Lytton, and really tell the story of how we got here. I want to go over what real world data is and what we can learn from it. And I want to get in and tell the story of this project, this data linkage of real world data and genomics in prostate cancer, our work in validating this approach and endpoints, and the actual study of a prognostic evaluation of this genomic classifier and some future directions. So I'll go a bit out of sequence, and usually we save these talks, these slides of collaboration and thanks for the end, but I think in honor of Dr. Lytton and the impact he's had here, I wanted to really highlight this. Really, this is how these stories happen, how everything comes from people. And I've been so touched by Dr. Lytton's legacy here. I've only got to meet him a few times at faculty events, but his imprint on everyone who worked with him is just extraordinary. I think of so many of my mentors and support here, Dr. Kohlberg, Dr. Weiss, or just in almost every interaction, talk about something Dr. Lytton said, something Dr. Lytton did. And so I'm very much imprinted with his legacy. And so that story continues in my own training and how we got to this story of this research project. I did my residency at Mount Sinai and was so lucky to work with Dr. Tewari at UCSF with Dr. Carroll and Matt Cooperberg, and sort of had me germinating these questions and thoughts of, how do we work with the data, with the information we have? How do we make better clinical decisions? At Yale, this story has been supported continuously by Dr. Kim, by mentors in the School of Public Health and Department of Medicine, Carrie Gross, Xiaomei Ma, Darryl Martin, John Onofre, Preston Sprinkle, people in radiation oncology, and through external collaborations. And I've been just incredibly fortunate to work with people such as Dr. Catalona, who are here today, an array of other collaborators as well, getting to this story. So the first question I want to begin to ask is, how and why did healthcare become data? We used to have these very straightforward interactions between clinician and patient, doctor and patient. And I think it's really out of opportunity and necessity that we started to think of healthcare interactions and healthcare delivery as data. And it really happened in two different ways. Number one, that necessity, right? We can make better inferences, better discoveries when we understand cause and effect. And so that is the necessity. And the second part is opportunity. As we've transformed how we deliver healthcare, as we begin to do it digitally and collect data that is coherent and able to be collected at a large scale, there is an unprecedented opportunity for discovery. And this is happening behind the scenes in everything we do through our electronic medical record interactions on Epic. Every little data point, every little thing you do is entered and tracked. And our insurers, the people who pay for it, keep exquisite records on everything we do. I think we all know that. What is real world data? Well, I think a convenient definition for today is it's any observational healthcare data that's not obtained in a clinical trial. It's what we actually do, the real world interactions we have, the vital signs we take, the laboratory findings, every service we bill for, everything we do is a real world data point. And we can collect and compile this to make inferences and about real world evidence, to make statements of understanding about how to actually deliver care and what things will result from a given intervention, exposure, and outcome. And it really is a broad array of different components. There are clinical pieces to it. So the clinical data that we keep, the insurance claims, every service we bill, financial data, clinical specialty data, patient satisfaction scores, and healthcare information exchange. But the goal has not changed. At the very elemental level, we're still trying to accomplish the same thing. We're trying to understand the relationship between disease, intervention, and outcome. That paradigm is unchanged, but I think we have a much more sophisticated way to approach it. And as one exemplar is the question of prostate cancer, right? We're still answering these questions, but how do we get from patient disease on the left, array of possible different interventions, including no treatment, surgery, intensified radiation, and androgen deprivation therapy, radiation, to affect the natural history of the disease and patient outcome. But there are obstacles to delivering precision medicine using real-world data, retrospective data. And I think there are really three I want to highlight and begin talking about some of our approaches to overcome these challenges. The first is that the disease is varied. It's incredibly heterogeneous. The patient biology varies, and how we deliver treatment is variable. So these are two very difficult variables to control for in all settings. Everyone's cancer is slightly different, and every treatment is slightly different. As much as we try to standardize them, they do change from patient to patient. And in this context, we speak about something called average treatment effect, which is when you aggregate the outcomes for lots and lots of patients, this is the average effect you see. But there's a lot that goes into that average. We are all sort of individual components here, and that average effect might mean something from patient to patient. The other challenge is that the data is messy, right? And so what does it mean to be messy? Well, there's a lot of things we don't know. The level of resolution that we measure in insurance claims or in cancer registry data is very poor. There is a big risk of unmeasured confounding, so things we cannot control for that obscure the true relationship between cause and effect. Also, our treatments might be outdated. By the time we're able to conduct a study, many of the interventions are actually well in the past. And so this is very apparent in cancer when we have a drug approved and we can begin studying its effect, but we're already on to the next one. And the last one is sort of a subtle point, is that in the future, as we are now more and more shaped by better prognostic estimates, by precision medicine, that information is actually altering what we do. So we are making inferences and statements about things that have happened in the past, but we are changing our behavior based on this information, which makes it more difficult to know how things will work, a bit circular. So about these, it's abundantly clear how varied prostate cancer is. And so these are just outcomes among patients with clinically localized disease, risks of recurrence stratified by CAPRA score, so based on simple clinical parameters. And so what's good for one patient is not necessarily good for another patient. That's very clear. The second question of limitations of retrospective data, and I think this is a good case study about making some of these claims. This is a paper that got a little bit of controversy looking at a comparative effectiveness of radiation versus surgery in localized prostate cancer using retrospective data from SEER and evaluating or estimating what the effect might be, but we can't control for a lot of variables. This is only what we know, right? We know that a patient had a certain Gleason score, a certain PSA level, and their exposure was either surgery or radiation. So there's a lot that might be missing in a story like this. And the last is the question of, well, how are we delivering on precision oncology? So the story that I'll be telling today is about a 22 gene genomic classifier, which is a commercial tool that many of us use in our clinic that allows us to deliver transcriptomic profiling, so essentially gene expression level information. And this is a very nice study published 2 years ago looking at stratification of risk among patients from a retrospective review of a clinical trial. So they basically took patients in a clinical trial, ran Decipher genomic profiling, and stratified the outcome. But we still don't know what happens in the real world setting. This is all data that's happened in the past, not in patients who've been treated prospectively. And so this vision for leveraging real world data and a genomics linkage is to unite highly detailed information about disease biology and outcome on a national scale, merging genomics data and clinical data. And so I want to walk through a bit of how we got here and what a data linkage looks like. And so at a very basic level, we are merging clinical data, so all of the data points that we see, patient interactions with genomics data. So all of the detailed reports and the full transcriptome of a patient that occurred through Decipher testing. And so working with Decipher, the company, we were able to take 95,000 full human transcriptome and merge those with a data resource of over 300 million patients. And the idea was to create a linkage where we merge the clinical information with the genomics information. So the first step was to identify both cohorts. These are completely distinct patient completely distinct. The first is this Decipher grid by Veracyte, so 95,000 patients where we had their de-identified grid expression signatures, their pathology information, the order information, and identifiers on them. And then link them with this pool of over 300 million patient interactions, including all structured electronic medical record data, claims data, and identifiers. And so question is, where does this come from? It's actually, these are all of the interactions that health insurance companies might have, that electronic health record systems have on patients. And using a secure tokenized exchange platform, begin merging the two. And so what this involves is sending secure encrypted tokens, basically identifying patients with a third party. So neither Decipher or Clarivate, who provides this clinical data, sees the information, but an anonymous token is exchanged between, allowing us to identify these patients and pull and merge the data. And in the final stage, we have all of the data, all the genomics data, all of the clinical data merged together, and we're able to successfully link over 90% of patients with full electronic health record data to their genomics data. And at the end, we have a complete clinical linkage. So once we've merged these two sources of information, the question is, did we do a good job? Is there anything meaningful or coherent that we can say with this information? So we went about a really painstaking series of steps to validate whether or not we are actually measuring the right things, because clinical data is messy. So the first thing we did is number one, wanted to see, are we actually accurately representing when a patient was treated and when a patient was diagnosed? And this plot here really is just showing concordance between when a patient was treated with surgery based on the clinical information that was filled out with a requisition to do genomics testing, with claims, with the insurance claims. And we found that there was a very good concordance. And so 98% of patients matched based on claim for radical prostatectomy and when the report was filled out for radical prostatectomy. It's a little bit lower for biopsy. There's a little bit more uncertainty there, but overall still very good. And so each cluster there is the year of treatment. And so we're hitting very tightly on each data point. The second point is, where are we getting this information from? And so these are the main outcomes that we're interested in. When was a patient diagnosed? Did they have a biopsy? Did they have a biochemical recurrence after their treatment? Did they develop metastases? These are all the relevant questions that we wanted to answer. For the majority, this is all identified from administrative claims codes. And so if we put in a claim, if you order a test, if you document something, and you use these terms, that's where we're getting the majority of it from. We're also getting some of this information from discrete data points, such as a PSA value. And this shows that step of PSA verification. So these are actual PSA data points taken from the electronic health record system. So this is part of that merger. And we're able to show that, number one, we can identify discrete PSA for a large number of patients. And there's also a correlation with treatment. So this is also kind of a sanity check to make sure that after a patient is treated, their PSA is dropping. And that helps verify that we are indeed identifying treatment. And we're seeing that. So that red line is the time of treatment, and PSA values go down. So suggesting that we're looking in the right direction. And when we finally merged it, this is sort of the beauty of it. This is the power of what we can see. So this is over 92,000 patients, transcriptomic profiling of their tumors and stratified by Gleason grade group. Okay, and it's pretty striking when you see the heterogeneity within each Gleason grade group. Okay, and on the y-axis, you see Decipher score, which is a continuous 0 to 1 score, 0 being the least aggressive, 1 being the most aggressive. And there's just considerable distribution within each of this. So leveraging this biologic information, this detailed report, we can begin to answer questions about the long-term clinical outcomes for each of these patients, for each of the 92,000 patients. And that's really the power and unprecedented ability to do something like that when you link clinical data and genomics data. So the first part was part one. How did we actually accomplish this linkage? And then the second part is what is the prognostic significance here? And so that's the second part of it. So taking this linkage of 92,000 patients, we wanted to answer a simple question. Is Decipher score significantly associated with those two endpoints, biochemical recurrence and metastases after treatment? We had to hone down that 92,000 patients a little bit to be a bit more confident that we're identifying patients who've been treated and that we have adequate follow-up for each of them. And we whittle that down to 58,000 patients, which is still quite a very large sample size. The patient characteristics, this is patients who've had biopsy and prostatectomy. The overall message here is they do fit the standard distribution that we would expect of patients treated in the contemporary era. Most patients had Gleason grade group 2 and 3 disease and were, sort of average treatment age. So this is really the first outcome, looking at the risk of biochemical recurrence after prostatectomy. This is again in that large cohort of patients treated. And it shows that there is a stratification. That Decipher category was significantly associated with the risk of recurrence after treatment, and in a multivariable model remained significant per 0.1 unit increase, adjusted hazard ratio of 1.14 per 0.1 increase in Decipher score. And that includes upon adjustment for significant clinical features such as margin status, seminal vesicle invasion, and lymph node status. We also saw a significant association between Decipher score and risk of metastasis after biopsy, including upon adjustment for all these factors, and also after treatment with radical prostatectomy as well. And so I'll stop there and just kind of highlight the main points here. Number one, that we successfully established the first national scale linkage of transcriptomic and longitudinal clinical data. And this was able to yield high accuracy for identifying key clinical junctures, including time of diagnosis, treatment, and early cancer outcome. This verified that the Decipher classifier was associated with the risk of biochemical recurrence and metastases following treatment. And I think it sort of opens an exciting door to leverage in the future to understand questions of disease biology, patterns of care, and treatment effectiveness. And so I look forward to keeping you updated on this. And so I want to thank everyone again, the collaborators from Veracyte, from Yale, and many other collaborators and supporters and funders who have supported this work. So thank you so much.
Questions?
Oh, yes.
Thank you, Michael. That was a great talk. I just am curious about the particulars of the clinical real world data.
Yes.
You had mentioned Clarivate, I think.
Yeah.
So I understand stuff on Epic. I want to know what happens, the data I put in Epic, how it ends up at Clarivate. And do the patients know that their data is being utilized? So you're not getting consent. I know it's de-identified like that token thing, but here we are sitting, and how does it get from point A, B, C, all the way to there? And do patients know about it?
Yeah, it's a fascinating question. When I learned about this, I was a bit alarmed because you're like, where does this come from? Clarivate, it's a bit opaque, right? A lot of this comes from insurance companies. Most of this is from the insurance company. So if you have Aetna, if you have Blue Cross, they hold every single data point, right? Every single interaction, every single service that they pay for exist in a database. And they are usually the ones that are sharing that data.
How do they get to the how does the insurance company get, we have CEWs, Epic, that's what.
Yep.
How does the insurance company get that data, get to their server?
Right. So insurance companies will not get the clinical data. So what Clarivate does is they aggregate and merge several sources. So one is insurance claims. So I think the lion's share we're seeing are insurance claims. And there are some EHRs that are sharing de-identified data as well. And they're getting paid for it.
So is there an ethical dilemma here? Another patient walking in. I'm not saying they wouldn't participate.
Yep.
It gets back to consent paper.
Yep.
Understanding what's happening with your data. You know what I'm saying? And so is there any talk about ethical construct of this whole thing?
Sure. Yeah. I mean, I think that it's, it's a great question, right? Patients are opting in for it. So it's just like when you click your data use agreement to use your iPhone or to use any product, I think it sort of falls in the same category that patients are agreeing to do so because it's sort of part of their, when they sign to get care at a certain facility, they're keeping that data. But you're right. I'm not aware of a strong movement to push back against this because it's almost impossible, right? The second you agree that we're going to use an EMR, right, that we're going to track these things, it's difficult to put constraints on what you're going to do with it, right? Hopefully a place like Yale would say, look, we're not going to sell this to a third party, but some of it comes through research, right? It's not purely a commercial enterprise. The overall goal is to make things better, but there are probably hundreds, maybe thousands of research projects just at Yale using Epic data, right? I imagine the same thing everywhere, right? Where we're trying to leverage all this data and study it. But how is it getting out? It's a good question. I don't think that major academic EMRs are selling it to places like Clarivate, but it's a bit opaque. We don't know. Yes, sobering thought. Yeah. Thank you. Thanks.
Okay, again, Dr. Leapman, thank you so much for sharing your research. So we'll move on to the next speaker, Dr. Mark Saltzman, another speaker who needs no introduction to our campus, but I'm just going to give you a brief overview. He finished his graduate training, doctorate degrees at MIT in chemical and medical engineering. He has been a faculty at Hopkins and Cornell before coming to Yale. He's also the founding chair of Yale's Department of Biomedical Engineering. Also noteworthy of crossing paths with Dr. Lytton is that he also served as a master at the Jonathan Edwards College. His research has impacted the field of drug delivery, biomaterials, and tissue engineering, and has published more than 300 papers to date. He has written 3 textbooks. Today, Dr. Saltzman will talk to us about bioengineering to treat cancer and improve kidney transplantation, standing on the shoulders of Dr. Bernard Lytton. Thank you, Dr. Saltzman.
Thank you. Thank you. It's a real pleasure for me to be here today. I'm a little nervous because I have used Dr. Lytton's name in the title here, and I want to live up to his grandeur. I can't hope to approach that, but I'll give it a try. So I'm going to talk about bioengineering to treat cancer and improve kidney transplantation. So I'll give you a little brief introduction and then talk about these 2 subjects and hopefully along the way explain the relevance of this work to my respect and admiration for Dr. Lytton. So I first met Dr. Lytton when I was invited to give a talk at this Fettschrift for Dr. Lytton that was organized by Dr. Weiss. So thank you very much for inviting me to that, Dr. Weiss, and give me a chance to meet Dr. Lytton and introduce him to the scientific work that we were doing and begin to develop the friendship that I am lucky to have shared with him. Now, I said standing on the shoulders of Bernard Lytton, but when I was first appointed master of Jonathan Edwards College, this was right after that appointment at the graduation in 2016, and I got to stand in front of him, which feels very inappropriate. But the rest of the time, I've been lagging behind. So bioengineering to treat cancer, and this really builds on work that Dr. Lytton recognized was important and long before I started working on it. And that's the idea of trying to administer chemotherapy focused at the site where chemotherapy is needed rather than allowing it to spread over the whole body. And in Dr. Lytton's approach, he infused chemotherapy drugs like 5-FU directly into the internal iliac artery to concentrate that drug at the site of bladder cancer. We took a different approach using biomaterials for releasing drugs slowly, but materials that could be placed at a local site. So this is a wafer. You can see it being held in the forceps here. It's a wafer made of a polymer. It's a degradable polymer. It degrades over a period of several months after you expose it to water or implant it in a tissue. And it would ordinarily be clear, but it's white here because there's chemotherapy drug dispersed throughout this matrix. And when you place it in water or put it in a tissue, that chemotherapy drug is slowly released over a period of time, here several weeks. So I developed this with collaborators whose names you see here, including Dr. Henry Brem, who led this study, chief of neurosurgery at Johns Hopkins now, and in collaboration with a pharmaceutical company. And the idea was to build this wafer as a tool that neurosurgeons could use when they did a resection of a glioblastoma. They could place the wafers in the resection cavity. They'd slowly release the drug agent, and that would hopefully kill any residual tumor cells that were in the vicinity of the resection site. And this is the result from the first clinical trial led by Dr. Brem, where you saw that patients that got a wafer that was loaded with BCNU lived significantly longer. And so this was approved by the FDA in 1996 and became a product that is available to patients with glioblastoma. And you can see here, this is work from my laboratory in an experimental animal with radio-labeled drug placed at a site in the brain, and you form sort of a ring of chemotherapy right around the site where you place it. So site-directed delivery of chemotherapy. We tried to take this concept and expand it to treatment of other kinds of tumors. And we did that in a couple of ways. One was by shrinking down the delivery system. So these are tiny, tiny particles that have many of the same characteristics of those wafers I just showed you. They're made of a degradable polymer, so they'll disappear over a period of time after introduced into the body. They have high levels of chemotherapy drug embedded in them, and so they can release them slowly with time. But they're very small. These are 100 nanometers or so in diameter, so the size of a virus. And these particles have some interesting properties, and you can see this in the graph here, that they're not toxic on their own, that drugs are slowly released from these materials over time, that if you put these materials in cell culture with tumor cells, the cells will be killed in a dose-dependent fashion. And sometimes, as in this example here, you put the drug and the nanoparticle is more effective than the drug by itself, which was a surprise to us initially. And if you inject these directly into tumors, they can halt the growth of even very aggressively growing tumors in animals. And we think these last two features are related to their size, the size of the particles. And you can see in this micrograph here, these are tumor cells, red outline, blue is the nucleus. The green are our nanoparticles, which are internalized inside the cells. And so they're slowly releasing drugs internally inside the cell, overcoming many biological barriers. And you can see some of the green particles are very near the nucleus, which is the site of action for many. Uh-oh, I did something to the screen here, but a familiar call for Jody. It didn't seem to affect the view here, but I don't know if it affected the. Yeah, it was full screen here before, so I don't know if that affected how it looks to the. Yeah. So this is our latest version of these nanoparticles. The orange part is where the drug is in the core, and that's the degradable polymer. The green coating is a corona around the green particle, which we've been able to chemically modify so that it's bioadhesive. When you inject these nanoparticles into a tissue, it adheres to proteins, cells very aggressively. And that allows you to inject them into a tumor or introduce them into a tissue space, and they'll remain in that tissue space while they're releasing the drug. It also increases their affinity for uptake in tumor cells. And we've used this with collaborators in a variety of different settings. One is as a local treatment for skin tumors, where we inject directly into squamous cell carcinoma, for example, or early melanoma lesions. And you can show that you get increased survival in these tumors in animals are quite aggressive, and they'll die if they're not treated. And we increase survival in that setting. You can inject them into the intraperitoneal space for patients that had, or in this case, animals that have peritoneal carcinomatosis as secondary to ovarian cancer. And they're very effective both at staying within the intraperitoneal space and killing tumor cells that reside there. And you can use them in glioblastoma as well, injecting them directly into the brain. And this is our latest study, which was just published a few months ago, showing that you can release anti-microRNA drugs into the brain and have a very profound effect on the development of patient-derived xenograft gliomas in mice. Bioengineering to improve kidney transplantation. Here also standing on the shoulders of Dr. Lytton, and this work has been referred to before, but I encourage you all to read this, this brilliant memoir of transplantation history at Yale and Dr. Lytton's role in it, and the role of many of you in this room. But he really had a multifaceted influence on transplantation, as many of you know, developing approaches for creating a fistula to allow the kind of long-term access that you need for dialysis that could keep patients alive while they're waiting for transplantation. He thought about the ethics of transplantation, something that continues to be a theme in transplantation research today. And as was mentioned earlier, thought deeply about the immunology of transplantation. One problem he couldn't solve was the problem that there's still more people waiting for kidneys than there are kidneys to transplant. And so expanding the pool of possible donor organs is another avenue that needs to be explored, and making organs better in terms of less reactive to the immune system is another potential innovation. What if you could change an organ so that the immune system of the recipient was less likely to reject it rather than treating them with immunosuppression? So one of the approaches for improving kidney quality, maybe rescuing marginal organs so they could be used as transplants, has been pioneered by Mike Nicholson in the UK collaborator, and Sarah Hosgood in his laboratory. And this is the idea of ex vivo machine perfusion before transplantation to resuscitate or improve the quality of organs. And if you have an organ that's on perfusion for some period of time before going into the patient, that gives you a second opportunity. And that is to introduce agents into the organ during the perfusion period. You can introduce agents like these nanoparticles that I've mentioned. And these nanoparticles could be targeted to the kidney endothelium. And it's the endothelium in the kidney that's the real site of action for the immune system. And if you could reduce the ability of endothelial cells to be recognized by host T cells, you might be able to improve transplant survival. So we undertook this program, and this is with Jordan Pober in the Department of Immunobiology here, a physician scientist, to test this idea that you could deliver nanoparticles into perfused human kidneys and deliver agents with them. Our first steps were to translate this from sort of a laboratory to the perfusion setting. And we did this by attaching anti-endothelial cell antibodies. So these are anti-CD31 antibodies attached to the surface of nanoparticles. And if you look at a plain nanoparticle versus one that has antibodies on the surface, you greatly increase the affinity of the nanoparticle to endothelial cells, as is shown here. But when we perfused these first into human kidneys, we found that the antibodies didn't provide as much of a benefit in terms of targeting to endothelium as they did in culture. So why is that? And this is one of our earliest studies where you see that there's red nanoparticles attached to the glomeruli, to the endothelial cells in the glomeruli and in the peritubular capillaries, but at relatively low levels, much lower than we expected. Two problems, if we want to expand this, if we want to hit nearly all the endothelial cells, which we suspect that you need to do, you need to solve, we think, two problems. One is that there's vascular obstructions inside the kidney that prevent access to nanoparticles to all of the cells. And the second is, as you can see from my diagram here, maybe the antibodies aren't oriented properly, so they're not working when they're on the surface of the nanoparticle. That first problem, that first hypothesis was really explored most thoroughly by my colleague, Greg Tijan, who identified that there were fibrin red blood cell plugs in many of the vessels in the kidney, and that this is related to cold ischemia time and age of the donor, but that those could be cleared by pre-treatment with TPA and plasminogen, and that you could clear those obstructions. And when you cleared those obstructions, you did a much better job of hitting all the endothelial cells with your antibody-targeted nanoparticles. The second approach was really a chemistry problem. How can we attach antibodies to the surface of these tiny particles in an oriented way? And we did this in collaboration with Shohei Koide at NYU, who developed these monobody adapters, the green elements that are shown here. You could attach the green element to the surface. That green element is a small protein that binds to the Fc region of antibodies. And when you then add antibodies, they line up in an organized way on the surface, and their antigen binding regions are now exposed. This is a bit busy, but it shows you a little bit about the chemistry of how we do this. Did it again. The chemistry of how we did this, attaching the monobody first and then the anti-CD31 antibody. But I'll just point out this cryo-TEM image on the right-hand side here. If you look at panel E, that's our conventional approach where you just see a glob of antibodies attached to the surface. And on the top, you see how organized the surface of the nanoparticle is. These antibodies are really standing on end all over the surface of the nanoparticle. It's quite remarkable. And this approach greatly enhances the binding of the nanoparticles to endothelial cells. You can see that both in static culture, in cells cultured under flow, and in isolated vessel perfusions. We get very profound increases in nanoparticle binding by organizing the antibodies in the right way. We tested these in perfusion of human kidneys using the approach I described before. Here we looked at a total of 6 kidneys, 3 that had the antibody nanoparticles, and 3 that had an irrelevant antibody, a non-binding antibody on the surface. And what you can tell is that we've done a much better job now of reaching these are biopsy sections from these human kidneys, low magnification and then higher magnification on the right-hand side, quantified in the bar graphs on the left. But you see now we're in the glomerulus, we're hitting about 70% of all the endothelial cells now have nanoparticles delivered to them. And in the peritubular capillaries, almost 50%. So we're getting close to where we think we need to be in order to really treat these kidneys before transplantation. What would we put in the nanoparticles? Well, there's a variety of things we could do, but we've been guided by the biology that Dr. Pober has identified. And one approach is to knock down expression of major histocompatibility proteins. And so in this case, we put siRNA directed against C2TA, a transactivator that controls expression of class 2 MHC on these cells. And this is done in a vessel that's implanted into a mouse with a human immune system. And you can see knockdown of HLA-DR 1 week, 2 weeks, 4 weeks, even 6 weeks after implantation of the vessel that was pre-treated before implantation with these siRNA-loaded nanoparticles. So I'll stop there by just thanking collaborators, and I hope I mentioned them as I went along, the ones that were involved in the studies I've shown you today. And just say a last word about standing on the shoulders of Bernie Lytton. I was fortunate to be head of college, the new name for masters of residential colleges here. And it's really a great privilege to be in that position. And this is a picture of Bernie with the class of 1994 who lived with him in the college while they had their Yale undergraduate experiences. At the reunion, and you see the familiar Dr. Lytton smile, delighted to be among these group of people that he really mentored and helped launch into the world. Thank you. They don't appear to. Most of what I talked about here, the first part, it was in the setting of cancer. So it's a little hard to tell in that setting because you've got so much going on in the tissue. If you just put them into normal tissue, inject them subcutaneously, inject them directly into the brain, we see very little response to the material itself. And we don't really see a response in the kidneys either. When you look closely at histological sections from those human kidneys, not much response. Of course, those are perfused and there's no other cells around.
Okay, well, we're right on time. Actually, we may be a couple minutes ahead. So again, thank you. I think speaks the volume about the quality of speakers that we have here this morning. So we'll move on to the next speaker, Dr. Vito Imbasciani. Dr. Imbasciani is also our alumni class, or he finished his training in our department in 1990, again with Dr. Lytton. He graduated from University of Vermont, Larner College of Medicine. And then after training at Yale, he wanted to have a 27-year career as an Army surgeon. And he currently practices urology in Southern California at Kaiser Permanente, and he has served as a secretary of California Department of Veterans Affairs since 2015. And most recently, he was appointed as the new chair of California Institute for Regenerative Medicine, which oversees about a $12 billion budget on an annual basis. So I'm delighted that Dr. Imbasciani is taking time to come out and give us a talk. And he'll be giving us his overview, or his topic is CIRM, a pioneer in regenerative medicine. Dr. Imbasciani.
Thank you, Dr. Kerr. Thank you, everyone. What an honor to be in the company of Dr. Lytton's biological offspring and everyone else in the audience. So we are his intellectual academic offspring. Very appropriate that I'm going to talk about a new job that I started 4 days ago. I got sworn in as the third chair of California's Institute of Regenerative Medicine, California Stem Cell Agency. Maybe the title is a little bit of a misnomer, because yesterday in talking to Yale's very distinguished faculty in its own stem cell institute here, Haifan Lin's accumulation of talent, I learned that maybe Connecticut was first. For 10 years earlier on, Connecticut funded at the government level, not like California out of bond authority, but out of Connecticut's general fund, $25, $50 million, something like that. It lasted for 10 years, but since it was general fund funded, when the state encountered financial difficulties, and around the time when the political parties shift, that support to Yale's effort ended. So I am going to introduce you to what I hope is going to be a giant step into the future. I am the head of a state agency. My new email is @cerm.ca.gov. It's a state agency that's unique in the United States and perhaps in the world. It was created to invest. There it is. To invest in California in 3 areas of regenerative medicine: research, education, and patient access. CERM was created through a citizen-led bond initiative in 2004 called Proposition 71. That initiative resulted from the desire of the people of California to pursue the potential for stem cell treatments. This was through a ballot initiative in 2004. If you'll remember what administration was in power in Washington at the time, an embargo was placed on the use of human embryonal stem cells at the federal level, and that had a chilling effect on research across the country. California tried to and succeeded in evading that by creating a safe haven. Proposition 71 amended the California Constitution to establish the right in law to conduct stem cell research in California. It created CIRM, and it authorized $3 billion in bond funding authority to fund this basic stem cell research. From the years 2004 to 2007, they were spent in battling lawsuits all the way up to the California Supreme Court. The plaintiffs in this case came out of the religious community and certain parts of the political spectrum in California that were hostile to the idea of using human stem cells for research. In 2020, as we got toward the end of that funding round, Proposition 14 was presented to the voters and passed again, giving CIRM an additional $5.5 billion. But it also gave me some stipulations. Since this is an amendment to the California Constitution, it's my law, I must follow it, but only the citizens of California can affect my agency, can change it in any way. So what I'm telling you is I have no government oversight, a $12 billion agency with no obligation to report to the executive branch or to the legislative branch, which is, I think, why the governor selected me to do this. So part of the mandates in Prop 14 are I must, in the next 4 or 5 years, dedicate $1.5 billion to neurodegenerative research, which I will expand to include neuropsychiatric and neurodevelopmental. I don't want to forget the children with autism and other pediatric issues. I must establish and build, CIRM actually builds, I'll get to it in a second, builds facilities on California medical school campuses to conduct this research, but I'm mandated to expedite the process by creating shared services, whether that's going to be machinery that different laboratories can use or a common bank of stem cells that can be available to all researchers. I also have to guarantee that the beneficiaries of this are not just rich white people from Beverly Hills, but I have to make in every possible way to reflect the diversity of the state of California, to make sure that the farm family in Fresno, the agricultural communities of the Central Valley, the people who live in the high Sierras, the Inland Empire, the high Mojave Desert, that they can all have access to our alpha clinics, which will deliver the care that are in our major metropolitan centers where the UC medical schools are. You'll notice in the title, regenerative was deliberately chosen so that it's not limited to human embryonic stem cell, but will include also gene therapy and small molecule therapy. Voters gave us our mission to accelerate. That's the word I like the best in my mission, to accelerate world-class science to deliver transformative, regenerative medical treatments in an equitable manner to a diverse California and world. CIRM is funded by the people of California, for the people of California, to advance regenerative medicine safely, equitably, and efficiently. We believe that regenerative medicine can address serious health issues that today have no known cures. I've listed some of the diseases here that we are funding. We are funding and supporting some of the best regenerative medicine researchers in the world. We can empower new generations of Californians to discover and prepare for careers. My final slides are going to dwell on this, and I think you'll be extraordinarily excited by this. So our strategic goals to advance world-class science Develop competency hubs. That's the function of what we call our originally 3 alpha clinics. They started in UCLA, UC City of Hope, and UC Davis. We've now expanded these are an alpha clinic is a FDA authorized state-of-the-art center for cell and regenerative therapy delivery. These are the places where patients go to enter our clinical trials to receive the regenerative therapy that has been designated for them to produce the therapy. So in a sense, the alpha clinics are also factories that will ultimately require a new workforce that has yet to be developed. We've expanded the alpha clinics now to UCLA, UC Irvine, UC San Diego, UC Davis, UC San Francisco, UC Berkeley. Notice that's where all the medical schools are, plus Stanford, which is outside the UC system, but has a medical school. USC Keck with its affiliation at Children's Hospital Los Angeles, the City of Hope, which is a cancer center outside of Los Angeles, and Cedars-Sinai Hospital in LA. Deliver real-world solutions. Part of this, you'll notice that the alpha clinics are all on the coast in all large metropolitan areas. I live in a city and county of 10 million people, but there are counties in California that only have 1,800 patients. So what we're doing, and I must create these by 2025, according to the law, I must create community care centers of excellence. These are places where we can spread awareness to the diverse communities of California of the existence of this, share information, recruit and retain patients into our clinical trials. I have to build them by 2025. We're in the process now of completing the last of our listening tours, Palm Springs, Inland Empire, Fresno, Humboldt, all the way up by the Oregon border. And we're going to devise another mandate in Prop 14. We've just created a committee of the board called the Access and Affordability Subcommittee of the board. We're going to find a 7-year-old boy who needs his sickle cell gene replaced by a working model of that defective gene. But that family lives in Tulare County in the Central Valley. This committee is going to figure out a way to how do we transport that child and his family and support them for the 3 weeks that they will be at Stanford or UCSF or Berkeley. The final bottom line principles of diversity, equity, and inclusion are so embedded in everything we do that when a grant proposal comes, for example, might come from one of the members of the urology department at Yale. Because even though this is a California initiative, if you have a program in any of your medical centers in the United States that can recruit California patients into your clinical trials, that's how I can support you. When those grant applications come to CIRM, they are scored by 2 different committees. One is a group of 250 scientists from outside California, even outside the United States, who are preeminent in cell and gene cell therapy, or genetics. They advise and score on a scale of 1 to 100, and then the application is given to patient advocates. These are not scientists. These represent the ALS community, the Alzheimer's community, diabetes, HIV, and also nurses who are expert in the conducting of clinical trials. They give every application a DEI score. And when both of those are scored and reach certain benchmarks, then those applications come before CIRM and I take them to the board. Here's a snapshot of our impact. We have to date funded 1,260 research and transformative programs. That breaks down into 658 in the discovery phase, 124 in the translational phase, 126 clinical grants. of which 91 are still active, meaning we have 91 active phase 3 clinical trials, 290 educational grants, and as I said, we build buildings and outfit our laboratories with infrastructure grants. They have 3,400 participants in our clinical trials, and I think I can just keep moving in the sake of time. This is our pathway. The path from stem cell therapy development to life-saving treatments can be challenging, but at CIRM, we've developed a series of funding programs that together improve the efficacy by which much-needed therapies reach patients in need. And that has a lot to do with these shared resources that I talked about that may be a strange concept to some people because a lot of laboratories are very protective of the intellectual property that is going on in their labs. But we've developed ways that whether it's shared cell banks or machinery to mass produce something, that can be shared. Well, just in 1 year, a snapshot, last year we awarded $347 million. This year, I just went to the treasurer of the state of California a month ago and said, this year I need $329 million. The treasurer sold the bonds. The money went to the controller of the state of California, who's my banker. In the next week or two, the controller will forward the $320 million to CIRM's bank, and then we will distribute that in regular payments to all the researchers. So we've put $62 million into basic science just in the last year, and so on. The educational grants come to in a second, $46 million to support the growth of our new workforce and interesting people that would otherwise never think of a career in science into just that, and $72 million into supporting the labs. Our initial investment of $271 a number of years ago, that went toward the creation of 12 buildings at UCLA, UC Irvine, Stanford, and Cedars. Those were built with co-funding. The co-funding has reached $62 billion coming from industry and the universities themselves, and to some small extent, something I want to augment, which is philanthropy. 50% of CIRM's research portfolio is invested in rare diseases, which is defined as the incidence of less than 1 in 200,000. But you can see that we are, even though I have a mandate of $1.5 billion to invest in neuro in the next 5 years, that we are already heavily invested in neurologic research, but there is very little part of the research spectrum that we are not supporting. Let me give you some examples. On the left, we recently invested $4 million in a late-stage preclinical project run by Ryne Bio, R-Y-N-E, to improve the treatment for Parkinson's. The proposed stem cell therapy will offer the potential to restore the dopamine neurons. So Ryne Bio is aiming to deliver dopamine-producing cells to replace the lost neurons to the brain of Parkinson's patients to restore and improve motor function. In the center is Karen Geisler from UCSF. She's working toward developing a safe and effective vaccine for acute myeloid leukemia, AML, to improve the relapse-free survival. AML, you all are familiar with what the disease is. About 20,000 new cases of AML are diagnosed each year in the United States, with a 5-year survival rate of only 29%. In 2022, there were 12,000 deaths in the United States from AML. Many AML patients, a majority of whom are over 60 years of age, relapse after treatment. To develop the cancer vaccine, Dr. Gansler and her team will engineer the patient's blood stem cells to maximize stimulation of leukemia-specific killing activity reintroduce the engineered cells back into the patient, which we hope will target and kill residual leukemia stem cells. And I'm sorry, what happened to our photos? Somebody should have said something. That's Dr. Gensler. And we have Evie Jr. On your far right. He's pioneering the search for a cure for sickle cell. Affects approximately 100,000 Americans, most of them Black and Latino. In July of 2020, he took part in a CIRM-funded clinical trial where his own blood stem cells were genetically modified to overcome the disease-causing mutation. Those cells were returned to him in one of our alpha clinics, and the hope is that they will create a sickle cell free blood supply. He has not visited the hospital once for any pain-related crisis or any other known manifestation of a sickle cell crisis since the transfusion. All of our clinical trials are designed to have follow-up on patients for 15 years. Cystinosis, you know, is a gene defect, single gene defect of the CTNS gene that causes the accumulation of cysteine in cells. You're familiar with the damage that can cause to kidneys, to cells. This is one of our patients in San Diego with his principal investigator. Jordan has to take 50 pills a day of cysteamine. The problem with cysteamine is it controls the cystinosis, but it accumulates in the skin and at a terrible price to this boy's social life because the patients give off this very intense smell of rotten eggs. Dr. Stephanie Cherky at UCSD, and by the way, there's some really wonderful videos that you can watch of these patients that give you a much more real flavor for this. She, in a phase 1/2 trial using CTNS corrected human stem cells, used human stem cell derived macrophages to transfer the normal protein back into him so that his tissues would no longer lack the protein. 4 patients have been treated. The first 3 of these patients infused with gene therapy remain off of their oral medication, both at the 12-month mark and 24-year mark. There's we can document the sustained engraftment in these patients, which is measured by vector copy numbers. The 4th patient was infused in 2021 with no adverse events to date. Using the same technology, Dr. Kuhl at UCLA has 50 babies that are bubble babies. These are the adenosine severe combined immunodeficiency. They're born with no immune system due to one defect in one gene. Using the same therapy at the UCLA stem cell, he's now cured 49 of these 50 babies. So no more immunocompromise. So I think even though that young fellow is only one of 500 Americans, that is a real orphan disease. The technology that was developed to treat him was then used to treat the ASA skid babies. I happen to have met Rosie Barrero. The Californians allow us to use their image and full names. They're very proud of their cures. She's 31 years old. She's blind since teenage years with retinitis pigmentosa. She has a 5-year-old daughter she's never seen. Dr. Henry Klassen at UC Irvine is an ophthalmologist, and using our support, created human progenitor retinal cells, injected them into her vitreous, and 6 months later, I can give you the science using the rat Schwann cells and how many of the transplanted progenitor cells are surviving. But anyway, she has vision. She can see her daughter now for the first time. She's not able to drive a car, but she can comb the baby's hair and pick out a pink dress. Okay, proof of concept. I'll settle for that, right? I hope the voters of California will settle for it when I hit them up with another bond 10 years from now. And finally, this is really remarkable. I just came just last week from UC Davis outside Sacramento. The head of surgery there, Diane Farmer, took our money, used an amniotic membrane patch and grafted placental cells on it. She's got 16 babies with spina bifida. Every urologist in this room knows, because every spina bifida goes through life with a urologist in their arsenal, right? So at 4 months of gestation in utero surgery, Dr. Fisher injected this dural patch, if you will. These placental cells are designed to recreate dura mater, which is the defect, what's missing in the final zip ring of the spinal cord. 3 of the babies have been born. They are moving their legs. So that's all I can report so far. These babies are normally born paralyzed from the waist down. I can't wait for when they're of the age to be toilet trained to see if there is any effect on bowel and bladder, right? But once again, proof of concept. So now this is the part I'm really, really happy about. You see the pictures here, that's California. We have a number of projects and we're funding it at the $100 million. We go throughout the state of California, find high school kids and put them into the laboratories of Stanford, UCLA, Berkeley, UC Davis, Irvine, all of our major medical centers, high school kids. We fund them to spend 8 weeks in these major research centers. And at the end, they come together in a symposium and they give their results. And to hear an 11th grader talk like he just spent 8 months in a Stanford lab, I mean, it's really remarkable. That's the SPARK program to spark interest. For the 118 2-year community colleges in California, we have a bridge program where we will take them in their to spend a summer. And on the UC campuses and the CSU, California State University campuses, of which there are 24, we take seniors and get them to spend 8 to 12 months, their entire senior year. Basically, we're paying their tuition and their upkeep to live in these and work in these laboratories of where all the preeminent science is being done. Those are the Compass and Bridges programs. We have programs to attract and support PhDs, postdocs, and our CERM clinical fellows. Look at the numbers. And these folks are writing peer-reviewed journal articles. I can tell you now the statistics at last review, 80% of these young men and women that reflect the diversity of California's populations have opted to pursue or remain in the scientific fields that they were exposed to as students. And I know this has barely been an overview. I'm sorry that I'm only 4 days into my new job. Maybe at some future point, I'll have a higher or more in-depth from the scientific point of view. I'm not a bench scientist. I am going to spend the next 3 months going to all the alpha clinics and to every one of the 91 clinical trials, learning as much of the science, because the governor picked me to make sure that I can make sure that every Californian knows where their money, I mean, California voters are on the hook to pay back these bonds, $12 billion in 30 years. Somebody has to make the argument, hey, folks, you made a great investment. But now we have wonderful stories to tell. You know, when I tell people these stories, I see them go like this. I know what this means. It means they got goosebumps, right? So, I hope that California will be, not to mix metaphors, a mecca for people, both scientists and patients in the near future to come to us for cure. Thank you.
Ryan's got a question.
That was a great talk. Thank you. I know you're doing on your job. Can you give me your projection where you think CERM's going to make an impact on urology since most of us do that? you have any thoughts about that? Maybe you need more time.
Well, Phil Kirshner over here, chief resident, we did tag. His last day was my first day, but we worked together for the last 20 years in Los Angeles. He was my chief of urology at our Kaiser Hospital. Serendipitously, he's come across a fellow at a university outside of California who's working on and has developed using stem cells a working kidney, a kidney that actually makes urine. And it was, come Phil.
And after I retired, I moved to Nashville. Met an oncologist who's been working on an artificial kidney for the last 20 years. His co-PI is Dr. Shubo Roy, who's a bioengineer at UC San Francisco. Vito and I, by chance, at dinner, went back to Los Angeles. Everybody was starting to artificial kidney. And I can tell you in the day, it absolutely works. It's got a microfilter. It's done like a transplant. You put a piece of ileum around it to drain it into the bladder. This has great potential. Great potential.
And to further expand on your question, there are 7,000 monogenetic diseases out there. That's the low-hanging fruit. You know, now that we've got the technology, give me somebody with a 1 gene defect, whether it's a cysteine stone former, okay, and we will start working on
it. Okay, so we'll take about a 10 or 15-minute break and they'll resume. Thank you. All right, again, thank you. I know that you know, it's just glad to be in one room again. I think pandemic also has taught us, you know, how important or how valuable these personal interactions are, these meetings. So I value that. But we do want to get going with our second part, so that we'll have many more another in the afternoon, the reception time to catch up. So the next speaker will be for this event is Dr. Darryl Martin. And Dr. Darryl Martin, you know, he, again, just to give you a background, he had a PhD from Memorial University in 2010. He's trained a background in cancer molecular biology and translational research in tumor growth and differentiation. And then after his doctorate degree, he did his postdoctoral training with Dr. Robert Weiss in our department, and then he subsequently stayed on to become a faculty member. He's currently an assistant professor in our department. And just to, I think, give you a direction of which direction that our department is taking right now, I think Dr. Martin really embodies the direction for our department in the sense of how we value science and basics. To that end, just to understand the fundamental work of how diseases occur in urology. So again, for a clinical department, it's oftentimes very challenging for us to have a basic science faculty member as part of the department. But again, it's just a testament to the commitment from our leaders as well as direction our departments going forward. In addition to Dr. Martin, we have 2 additional basic scientist faculty members that's coming on, or 1 has already joined, 3rd one's about to come on in the summer, as well as currently recruiting a 4th basic science faculty member. So again, this is just a testament to the direction and the commitment of Yale Department of Urology and moving our field forward. Enough said. So Dr. Martin, can you come and he will be speaking about the developments in prostate cancer research. Thank you.
Good morning. I'd like to thank everybody for having me here. How about now? Here now? Okay. So I'm a basic scientist by training from Newfoundland, and I was brought here by Dr. Weiss a number of years ago now, and to work on bladder cancer and drug delivery studies. And we worked closely with Mark Saltzman, who you heard speak earlier. So it was a privilege of mine to get to work with Mark also, as well as Dr. Weiss. Today, I'm going to give some background of some current work I'm doing in my lab now that we're branching out a little bit and doing some stuff on prostate cancer. And today's talk is really what I'm interested in is metastatic prostate cancer and really the role of testosterone in that. So just a brief overline of the biology, some that I'm going to talk about, the mouse models, the metastases that we're looking at. Again, this bipolar androgen therapy and how it may play a role, and then a few future directions where we hope to go. So mainly here, you're looking at prostate cancer, 288,000 estimated new cases this year, 37,000 deaths. The most important is really you're looking at really the 30% survival for distance disease. And that's, prostate cancer outside the gland, so the liver, bone, and lung. And of course, there's currently no treatments that can cure advanced prostate cancer. So really, I'm interested more in the biology behind it and how we can look at the metastatic disease. So one thing we look at is transcriptomics, and you heard Michael give a great talk earlier on transcriptomics. And we're also using transcriptomics in the lab to look at human tissues and try to figure out, looking at these RNA transcripts, how they change across different conditions and what we can do. So we basically take a sample from the patient, we collect it, we run the transcriptomic part, and then we get the outcome. This is all based on tissue-based molecular assays, and which are also part of the NCCN guidelines right now. So these are available tissue-based tests for prostate cancer, looking at risk stratification and prognosis. And as you can see here, we have on the left, the tests could be Decipher, as was mentioned earlier. You have Oncotype DX, Polaris. These are just different versions of transcriptomics or signatures that you could use. And these signatures really work best or are recommended for NCCN low, very low, or favorable intermediate disease risk in patients with at least 10 years life expectancy. So looking at transcriptomics, one of the ones we looked at, of course, is Decipher. As Mike mentioned earlier, is based on 22 gene expression profile and includes genes such as cell proliferation, cell structure. You're looking at cell cycle progression, androgen signaling, and this helps predict what the chances are of a certain tumor to metastasize in the future. So this is a commercially available signature. Another one that's not commercially available is Galinsky that was published, and this is based on 11 RNA genes, and this has a different profile. It's based on stem cells, and they also predict distant metastases. So what we are doing and currently doing, we're taking these different types of signatures that can predict metastases and trying to determine in our cohort, so we have a small cohort of patients here looking at low risk versus high risk, and just to show you some outputs, basically to show the same trend with two completely different transcriptomic gene signatures. So what we're trying to do is look at which profile or which signature will give the best outcome depending on the situation. And of course, the Galinsky here, we have high risk patients showing high proliferation score, and again, low risk showing a low risk proliferation score. So that's what you would expect. And again, with Decipher, we have high risk for a Decipher score, and then the low risk has a lower Decipher score. So these are what we would expect of happening. So now what we're trying to do is, again, combine either these same transcripts or same genes or combinations of multiple ones to see if we can get better insight into the metastatic nature of the disease. In addition, looking at human tissues, we also look at mouse models. And some of the work we're doing is we can look at genetically engineered mice. We can look at pseudo-metastatic models that we have, or patient-derived models. So they all have their positive aspects. Genetically engineered, of course, you can knock out specific genes, which are great. The drawback to this model, though, is, of course, it's the lack of reliable metastases to bone. So for prostate cancer, that wouldn't be, in most cases, a great model. Also has limited human disease relevance. We also have other models that we could inject cells directly into the bone if we're looking at a secondary site. But in this type of model, even though we can have different sites, you're kind of bypassing several important steps during the metastatic pathways. So this, again, wouldn't be ideal model if you want to look at how metastases happen, form, and how they progress. The last model I'm going to talk about right now is the patient-derived model. This is a great model to have human tissue we can put into a mouse as a vehicle and basically look at the growth to study the tumor itself. The only drawback from traditional PDX models is they're immunocompromised. But that being said, the PDX is where we decided to go, and I'll give you an example. So this is a tissue that a patient that we secured tissue. It was from a radical prostatectomy. This patient had a Gleason score of 4+4, or grade group 4, cribriform pattern. It had 10% of prostate involvement. There was not identified of urinary bladder neck invasion or cervical invasion. Looking at the lymph nodes, examined 33, and there was no involvement of lymph nodes. But so with this tissue we had, we also looked at from here the pathology and histology of the tissue. And as you can see here we have outlined in the dotted line here in black. Here is the region of interest we're interested in. This is the 4+4 high grade of this patient tissue. So we would use this tissue to move forward to make our models. So with metastases, just to take a step back, the percentages of metastases of local prostate cancer is about 14% for grade group 3, 24% for grade group 4, and about 41% for grade group 5. And where do you normally find these? Primary sites include liver, lung, bone, and lymph nodes. So that would be a model that we'd be more interested in trying to achieve, something that more recapitulates what happens in a human. We're trying to do that same thing in a mouse model. And once the cancer, of course, metastasizes, it's more difficult to treat and the prognosis is generally poor. So that's the first model we're setting up. We got, from that patient, we have the tumor. We set up the mouse model. Again, this mouse has no B or T cells, so there's no influence from the immune system with that respect. And it's a good way to right now to look at the efficacy of a drug on the tumor. So from that, we ended up growing this mouse, and this is one of the mice that grew a prostate PDX tumor. And as you can see here, this is just a sheet of cancer. So it worked out well. We have a high grade back. We put high grade in, we had high grade back. So our mouse model was effective in what we were trying to prove as a first step. But the real goal here really is to move into and not just create a PDX, but a metastatic PDX, something that we can actually use that would be more useful. So with this model, we looked and actually developed metastases to two distant organs. And again, as I mentioned, this replicates the most lethal prostate cancer if it can hit those major sites. So just to show you the first one, when we examined the mice, the top is the cartoons for these. Of course, the bottom is an H&E, and around you'll see a dotted line around the cancer. So this is with the patient PDX we had. This is the first one. You can see that this is lung. So we have prostate cancer that metastasized to the lung in this model. We also had bone. So it actually metastasized to the bone, to the liver, to the kidney, in this case, the secondary site, and then of course the lymph node. So our mouse is able to generate or develop metastases to the major areas that really would recapitulate what's happening in the patient. So it was a better model to start to study, to look at, to understand how we can manipulate this and to look further at the molecular aspects. So also, as I just showed you, that was H&Es from the mice. This is another H&E on top of the kidney with prostate cancer. So now we just had to prove that this was actually prostate cancer, not mouse cancer, which could happen. So we actually did, the bottom is going to be immunofluorescence, that we have green, going to stain for an antibody to stain mouse cells. Then the blue is going to be DAPI for nuclear stain, and the red stains the human cancer. So now, as you can see, all the metastatic sites were actually human cancer that's metastasizing to those sites. And as the next confirmatory part, we just looked at some markers that we're looking at, PSMA, as well as Claudin-4, a tight junction protein that we found in previous studies to be highly upregulated in some prostate cancer models. And in our primary sites, we found that 2 of these have been upregulated, as well as the metastatic site. We still had high expression of both of these biomarkers. So from here, the story changed a little bit. So now we had a model. And as I mentioned before, in generating this model, we had been a very difficult model to generate, prostate PDX. We tried a number of things to try to really amp up and make the cancer more aggressive. And one of the things we thought at the beginning when we're trying to develop it was giving a high doses of testosterone. And just from what you would expect you would think that high doses could link to an advantage for growth of prostate cancer. Other studies have not found a clear association between testosterone levels and prostate cancer risk. But if you look at the standard treatments for patients with high or very high disease based on NCCN guidelines, androgen deprivation theory is the first line of defense for treatment. And basically you're really just reducing the levels of androgen in the body, androgens, and it blocks testosterone production. So again, this made it seem like this would be a good avenue. Either you really upregulate the amount of testosterone to make the cancer more aggressive. But ADT is commonly used and it really can help shrink the tumors before radiotherapy. There's some disadvantages, loss of libido, erectile dysfunction, and hot flashes. But looking at a cartoon form, you have tumor growth at the beginning, you get undergoing local therapy. It kind of comes back to prostate. You block it or knock it down with ADT. It comes back or reduces at that point cancer, and then eventually that you will see it'll undergo metastases at some point in the future, become resistant to ADT and undergo metastases. So this was intriguing of what happened here and why testosterone in our case was not really making the cancer more aggressive. So that's when we're looking at bipolar androgen therapy, or BAT, and was pioneered by Samuel DeMeade from Hopkins. And the strategy was really looking at a variation in giving supraphysiological and castration levels at the same time. So really you're exchanging high levels and low levels of testosterone. And this was supposed to be used to treat metastatic cancer. And it's really counterintuitive because, again, as I mentioned, testosterone to treat it seems like it wouldn't be the right thing to do. But there was preclinical models that shown that BAT could cause or at least reduce regression of tumors. There was a few small clinical trials that said that it was actually effective, and for ones that stopped responding to other hormones therapies. And so our goals then were really to look at who are the responders and non-responders here, how often is it needed, or the cycles. Is there long-term safety and efficacy concerns of going on high doses and low doses of basically testosterone? So for us, the first thing to do is we had to go back to our model and really figure out what was happening, and could testosterone actually be doing opposite what we thought originally. So we have the same mouse model again, no T on the side, you can see here. And when we give it high doses of testosterone, you can see in the lung, we actually resolved prostate cancer metastasizing to the lung, to the bone, to the liver, and kidney. So really regressed and wiped out prostate cancer in all those areas that we've generated. It did not impact the lymph node as of now, but it shows that high doses appear to be working. So then we went back and looked at the tumor volumes in these mice. And again, it's trending that no testosterone had higher levels of tumor volume compared to the high T ones. So we also looked at the metastatic sites. So what we found was that we came up with a MET score. So basically where the cancer metastasized to, and then how much of the organ was engulfed in the prostate cancer. And the mice that had no testosterone had much higher MET scores compared to those that were on high T. So really then we're looking at the beginning, we were probably suppressing our mouse model and not even knowing it by giving a high dose of testosterone. So now we actually have a model that we can actually create METs and kind of suppress the METs at the same time with manipulation of testosterone only. So right now, as of yesterday, we just started transcriptomics. So I unfortunately don't have any data to show you on this part, but we're looking at clustering and looking at which pathways or genes are actually responsible. So we can actually see if there's a biomarker or some type of whole pathway that we could actually target and find out what's causing this. So that'll be hopefully in a future talk I can give you guys. But for right now, the next directions of what we want to do with these mouse models is we want to track the metastatic tumors. We also want to look at the tumor immune interactions in the microenvironment. So one of the things we're doing right now is from our mice that we actually generated cell lines, high T and low T or no T cell lines, PDX cell lines from these mice. And we're actually now in the process of transforming these cells with a GFP marker or some luciferase so we could actually tag and find out where these cells are actually going first. So again, here's our prostate mouse, and we're going to get some tracking and find out where it's going to go. So that's our basic model, what we're going to hope is going to happen here. In addition, as I mentioned, one of the main parts of this was the PDX. And one of the downfalls, of course, was there's no immune, it's immune compromised. But back at the beginning, some of our groundwork when we're planning this was not just to have a PDX, but our vision was to have the humanized PDX to really be able to play on that aspect of what's happening to these mice when the immune system is involved. So that's why we actually now could turn our mice into humanized mice. So we just don't have PDX mice, but humanized mice. Now we can actually answer other questions. Does testosterone impact or activate any immune pathways? And how can these interactions of bat and immune be investigated? So now we actually have the model to do this. So as a first step, here's our humans with their blood and their tumors, and we generate the mouse models. The y-axis here is mouse CD45, and you can see that's going to be in any mouse. That's the presence of CD45. As on the bottom on the x-axis, you're going to see human CD45, and that's when we inject human immune cells from the same patient as the tumor. And now you can see we have a wild type mouse that has no human CD45, whereas the humanized mice has an ample amount of CD45. So we know that our mouse model is actually active and working at this point. The next step, of course, is to take this mouse model now into, we're looking at different drugs that could impact the immune system, or looking at go back to testosterone again, and just as a first step, see what's happening at that stage, any impact on the immune system or tumor response. So this is our little model that we're looking at, that high doses directly on CD4, CD8 NK cells as a starting point. If we can see if there's any impact there first, or if it's just strictly on the prostate. And we're trying to just figure out which pathways are going to be involved. So that's where we're going to bring back in transcriptomics on these mice. And hopefully with some of this data, we can actually see what the next targets we're going to have. So I'd like to thank some of the people that were involved in my lab, Stephen and Brian, and of course, Peter Humphrey and Preston Sprinkle were instrumental in helping me collect the tissue and get this to the lab. And of course, lots of great talks with Dan, Isaac, and Al Botwell to help with my mouse model. Of course, had to shout out to the GU residents for all the help of collecting tissue, sample consent, which is you absolutely need, and of course, a mentor, friend, and colleague, Dr. Weiss. So thank you. And have any questions?
Question? Okay. What's the genetic strain of your mouse that you're putting the tumor in?
This is it's like a skid mouse. So compromised. Yeah. So that's why I said no B cells, no T cells in this one.
I must have missed that. Thank you.
Yep.
So that's background. If you take a patient's primary prostate cancer, their transcription from the metastatic cancer is very different profile. How different is it if you take that patient's metastatic cancer, put it in another animal like a mouse that's immune compromised? How much does it change?
I mean, so you're just talking about how much would the metastatic change to another metastatic sort of just change in general?
The transcription profile, the proteins in the memory of that biology tumor in the human.
Right.
That same tumor then going into a mouse. Does it change?
Well, that's kind of what we're looking at right now. And so for that one patient that we had, that we worked backwards. So now we have the patient's transcriptome done, and now we're doing on the mice. So we're going to cross and look at what's different. So our goal is hopefully it'll be the same, similar pathways that we're looking at, but I don't have all that data at the moment. I would assume that it would be fairly similar at this point, but there's going to be changes along the way. I mean, the tumor is still evolving and still growing. So it's, just like in a patient, you're not going to, at your primary site and your metastatic site, they're not going to be the same, as you mentioned. So how much more involvement does it get? Probably depends on how many more generations we can get the mouse also to keep growing.
When you're getting the mice to the supercritical testosterone levels, how high are you going? Or what levels are they getting to?
Right now, we're probably 10 times higher. So we're really amping it up pretty high. And we're actually trying a whole bunch of different things of even low dosing, lower doses, half doses of that 5 times. Really just depends on how much we can get into the mouse safely. And and that's just one aspect of dosing up front. And then can we dose it once the tumor is already there? I mean, there's a whole bunch of different trials we're trying to do with it.
Thank you. Okay, we'll move on to the next speaker, Dr. Robert Weiss. Again, Dr. Weiss also needs no introduction. He's our former chair. But just to give you again, because I don't think a lot of our residents know exactly where, some of the details. So anyways, with Dr. Weiss, received his medical degree from the State University of New York and downstate. Then he went on to do the residency training at the Presbyterian Hospital, Columbia. Then he came on to Yale to be our faculty member, and then he became the chief, and he's here today. So Dr. Weiss will be speaking to us about his research. Again, this is a testament to who he is. While he's a consummate surgeon, also he's a scientist, and this is something that he's been pursuing for a long time in various areas of urology. So this morning, the topic of his talk is functional siRNA-loaded nanoparticles targeting urologic diseases. Dr. Weiss.
Dr. Kim asked me to give this research talk here today for this symposium honoring Bernard Lytton, Dr. Lytton. Successful research depends on several things. One is picking a proper mentor and environment to do the research. And that was my business of becoming to Yale and working with Bernard Lytton. None of this could have occurred without that collaboration. Selecting excellent colleagues, and this research could not have been done without Marshall Wheeler. Asking an important question and then enlisting collaborators who would be able to help in answering the question. And that would not have been possible without Mark Saltzman and his group in the Department of Biomedical Engineering. And finally, selecting a mentee to bring the research to fruition and to advance the science. And that was Darryl Martin, who is now an independent investigator who you've just heard speak. However, although this was an excellent team, they couldn't solve every problem. This research was supported by grants from the NIH and the Yale Cancer Center. In the early 90s, in really the late 90s, we showed the presence of an anti-apoptotic protein highly expressed in high-grade bladder tumors, and that protein was not seen. That protein was not seen in normal tissues nor in low-grade bladder tumors. And that paper was published in the New England Journal of Medicine. Hubert Swanner at that time was a resident. He's now a pediatric urologist, and he's here with us today. Survivin is a 16.5 kilodalton protein, which is an inhibitor of apoptosis and is located on chromosome 17. It's abundant during early embryonic development, but absent or present in very low amounts in normal adult tissues. It's highly expressed in most cancer cells, and it's associated with a decreased survival and increased aggressiveness. Shannon Smith, in a paper published in JAMA, showed that there was a 259 base pair survivin particle in the urines of patients with transitional cell carcinoma of the urine, and that was not seen in controls. Also, the beta-actin is a control that you see here. So knowing that survivin is highly expressed and related to aggressive tumors. The next thing, which was obvious, was let us try to inhibit survivin or to at least downregulate the survivin. And at that time, there was no small molecule, non-toxic inhibitor of survivin. So we considered the possibility of using sRNAs as an option to downregulate the survivin. sRNAs efficiently degrade specifically targeted mRNAs. They have a high degree of sensitivity and specificity because of their robust gene selective silencing of targeted proteins. They are safe since they do not integrate into the genome, and they are of relatively low cost and relatively easy to produce. The first thing we did was we showed that indeed we could use survivin sRNA to downregulate survivin mRNA in the red and survivin protein in the black. A scramble had no effect. Although sRNAs have an advantage, there are multiple disadvantages, such as their short half-life, their poor intracellular uptake of naked sRNAs because of their negative charge, and their vulnerability to degradation. At that point in time, if my recollection is correct, we knew that Dr. Saltzman and his group at the Department of Biomedical Engineering were successful in encapsulating many DNAs, proteins, drugs, and at that time, I don't think that anyone had encapsulated sRNAs, and we went to Dr. Saltzman and presented this problem, and he was successful in helping us to encapsulate the sRNAs into nanoparticles, and as you'll see, that's what gave it the possibility of treating various diseases, including bladder cancer. The nanoparticles have the advantage of in the administration of drugs and sRNAs, since they protect from degradation, they allow for slow release of their cargo over time. They increase functional half-life. They permit endosomal escape. They facilitate combination treatment if you wanted to use two drugs or a drug and an sRNA. They help in the targeting of specific cell types, which was mentioned before, and they facilitate tissue penetration. The PLGA nanoparticles that Dr. Saltzman showed you are biocompatible, biodegradable polymers approved by the FDA for the use of delivering of drugs, DNAs, peptides, and proteins. And Dr. Saltzman and his group helped in arranging it for encapsulating sRNAs and delivering them. They can be modified, which is important, and that's what I'm going to be showing you to increase targeting, uptake, and endosomal escape. The nanoparticle that we've used most often is the PLGA nanoparticle. The cargoes that we've put into it are chemotherapeutic agents, sRNAs such as the survivin sRNA, and I will show you later the GP130 sRNA. We've encapsulated fluorophores and iron, which has been used for detection with the MRI. On these nanoparticles are placed the anchors, avidin and PEG, which allow for the attachment of surface modifiers, chitosan and pgon, which functionalize the nanoparticles and aid them, and I'm going to get into that. Chitosan is a mucoadhesive polysaccharide. It's derived from the exoskeleton of crustaceans. It lacks mammalian toxicity, and it facilitates the penetration into various tissues. Pgon also has been developed as an antimicrobial polymer. It lacks toxicity, mammalian toxicity, and its cationic properties help to deform and penetrate the urothelium and other tissues. The nanoparticles range in size from about 75 to 400 nanometers. and the mean is about 150 nanometers. And the important thing from this slide is that if you surface modify it, which we mentioned with chitosan, you do not change the size of the nanoparticles to that extent. This immunofluorescent study shows the uptake onto and into T24 bladder cancer cells in vitro. And the surface modified ones are the ones to the right, Pgon, chitosan, and enteropedia. And one can see in the untreated cells that the nucleus stains blue and the cell membrane red. And following 2 hours of uptake of these nanoparticles, which contain coumarin-6, so they can be visualized, you can see that there's a much greater uptake using the surface modified nanoparticles than you see when there's no targeting protein present, such as the avidin. And then what we did is to wash off the nanoparticles from the cells. And you can see that after that period of time, the nanoparticles are in the cytoplasm of the bladder cancer cells. And it's primarily in the cytoplasm of those cells which have been surface modified, and very little among the avidin ones, which are not surface modified. You see very little of the nanoparticles within the cell. This just shows again that if you surface modify the nanoparticles, they're taken up into a much greater extent into the human ureteral segments in vitro and to the mouse bladder, which was done in vivo. These are step section fluorescent images of the mouse bladder. The nanoparticles were placed in the top, way on the top, and allowed to stay inside the lumen of the bladder. is the urothelium, LP is the lamina propria, DM is the detrusor muscle. The nanoparticles on the left were just empty nanoparticles. They're not surface modified. The ones in the middle are also a control. They have the anchor, but they're not surface modified. And the ones on the right have been surface modified. And what you can see is where all those white arrows, that's where the nanoparticles went. And only in the surface modified nanoparticles do they go down the various layers and wind up in the detrusor muscle itself. And this is just a magnified view of this. This shows prolonged knockdown of survivin expression over 9 days in UC3 bladder cancer cells with chitosan surface modified nanoparticles containing survivin siRNA. And you see only in the surface modified, the ones with chitosan all the way to the right, is there a down regulation in survivin. The others, even though they contain the survivin siRNA, do not actually release it and download, regulate the survivin. The type of surface modification affects the release from the nanoparticles. And these are just 2 different types of surface modifications and you can see that they're totally different over 2 weeks, where the survivin siRNA is released into a artificial urine environment. We then had, as Darryl mentioned, we're interested in we can work on the survivin siRNA in cells. Will it decrease the tumor? So a transgenic mouse model was made by the injection of 10 to the 7th UC3 bladder cancer cells into the flank of female nude mice. And over time, because the cells were transfected with a red fluorescent protein, we could see the size of the tumor, and also we could use calipers. And this shows in the growth of the tumor over time. You had various tumors. The zero date is about 7 days after the injection of the cells, when the tumor is now about 100 millimeters cube. And what you see is that only the blue line, only those tumors where we injected, and they're injected where the arrows are, at 0, 4, and 7 days. Only, I'm sorry, well, I guess it's okay, there it's okay. Only the blue ones, only the blue side, which had surface modification and the sRNA in it, did not grow. The virus controls grew well. Over time, the tumors treated with the nanoparticles containing survivin sRNA, it exhibited a 65 to 75% reduction in survivin expression compared to the controls. Thus, we have shown that surface modification of the nanoparticles encapsulating survivin sRNA facilitated the uptake and penetration of in vivo and ex vivo benign urinary tissues and into bladder cancer cells. Surface modification also decreased the survivin expression and tumor growth of the UM-UC3 cancer xenografts. We then looked at another sRNA to see whether it was involved. Anyway, this one was GP130, and to see whether it was involved in tumor growth. Inhibition or activation of a number of signaling cascades, including the PI3 kinase mTOR cascade, the MAP kinase ERK cascade, or the JAK-STAT cascade, is involved in development of resistance to chemotherapeutic agents, inhibition of apoptosis, angiogenesis, tumor proliferation, tumor invasion, and metastasis. As GP130, a receptor responsible for transmitting the signals of IL-6, which is involved in many aggressive bladder cancers and other cancers, is located at a central point of a number of these oncogenic signaling cascades, which I mentioned. We postulated that if one inhibited GP130 with GP130 sRNA, we would decrease tumor size. GP130 expression is greater in high-grade than in low-grade human bladder cancer specimens. GP130, as expected, is higher in tumors than in normal tissue, and you can see that survivin is also greater as expected in tumor than in normal tissues. GP130 expression is higher in more aggressive human bladder cancer lines than in the lower differentiated ones. And RT4 is a low grade, which we used to call a papilloma, grade 1 transitional cell carcinoma. And you see that it doesn't have very much expression of GP130. This shows that if you inhibit the GP130 with GP130 sRNA in two different types of cells, bladder cancer cells, you can decrease the viability. This is a cell growth assay using crystal violet, which demonstrates that decreasing GP130 expression with GP130 sRNA decreases the growth, cell growth, compared to the variety of controls that you see up there. They're all, the other three are controls. We also looked at a cell migration assay. In a cell migration assay, we took the bladder cancer cells which were the UMUC3 bladder cancer cells, put them in a petri dish, and allowed them to grow to confluence. And then at that point in time, made a scratch in each of the petri dishes. And what we can see is that by 12 hours, although the bad cells, the cancer cells, have covered most of the scratch, in the group that the GP130 is downregulated, the ones with the survivin GP130 are not covering it as well. And at 18 hours, the controls are all confluent again, whereas there's still a slower growth in the cells which were treated with GP130 sRNA. So knocking down GP130 expression results in cells that migrated at a slower rate than the untreated controls. We made the same type of xenograft mouse to treat them with the GP130 sRNA. And what you can see is, again, 0 is the time of starting to inject it, that only those tumors in which we injected them with surface modified siRNA containing nanoparticles had a decrease in growth. Even like the surface modified nanoparticles in red, where they don't contain the survivin sRNA, but they are surface modified, do not affect the growth of the tumor. This shows that after 2 weeks, the tumors in mice that have been treated with the GP130 sRNA are smaller than those of the others. And in concurrence with IRB regulations, all mice were de-identified and anonymity was maintained. This shows that the tumors were small, but in addition that the tumors being small, there is a decrease when you use a survivin sRNA containing, not survivin, GP130 sRNA in the nanoparticles. There's a decrease in GP130 expression and also a decrease in the phosphorylation of mTOR and AKT, which are downstream proteins involved in the signal transduction pathway with respect to tumors. So thus, sRNAs possess a high degree of sensitivity and specificity to degrade specifically targeted mRNAs. They are associated with advanced bladder cancer, and those we can downregulate with the sRNAs. Survivin and GP130 sRNA decrease survivin and GP130 expression respectively. They also decrease cell viability and cell migration. Incorporation of survivin or GP130 sRNA in surface modified nanoparticles facilitates the delivery and prolongs the release in sufficient quantities to downregulate the targets in vivo tumor models and inhibit tumor growth. I thank the collaborators, which otherwise this research could not be done. Christos Kamikliotis, Shannon Smith, and Hubert Swanner were residents at that time. Thank you.
All right, so we'll move on to the next speaker, another giant in our field, Dr. William Catalona. Dr. Catalona, a physicist from Chicago. I know that he had a long ride yesterday coming from the airport to here, but again, Dr. Catalona, we're really, really grateful that you're able to join us today. Dr. Catalona went to our medical school in 1968, or he graduated from there. Then he subsequently, after an internship at Yale, then went on to Johns Hopkins for his residency training. He's currently at Northwestern University, a professor. He told me yesterday he's still performing 3 OR days a week. It's amazing. He has performed over 8,000 radical prostatectomies to date. So, and for our residents, he's the one who showed us that PSA is the one that we need to use for screening prostate cancer. So again, a giant in our field. delighted that he's here, able to join us and give us talk on, let's see what the title is. I think factors associated with conversion from active surveillance to treatment for prostate cancer. Thank you, Dr. Catalona.
Thanks, guys. Hey, can you hear me? So I was, first I'd like to thank Dr. Kim for inviting me to participate in this memorial service for Dr. Lytton. I was incredibly lucky that fate assigned me to Dr. Lytton and as my faculty advisor when I was a first-year medical student at Yale. As everybody said today, he was a charming, charismatic person. He was a wonderful role model, and actually, it was he who inspired me to go into urology when I was a medical student. And we remained colleagues and friends ever since that time. And we were at a meeting with the Weisses a couple of weeks ago, and we were commenting to each other how much we miss sharing the good times at the meetings with Dawn and Bernie. So I'm going to report on a study that we completed about a year ago on factors associated with conversion from active surveillance for prostate cancer to active treatment. And I'm going to talk about two types of factors. One are the clinical factors, and the other are the germline genomic factors. Now, we've heard about the Decipher test, and so that is not a germline. That is not a germline test. You're taking the tumor sample and you're sending that for genetic analysis. So what we're talking about here is the germline genetics that exists in all of the cells in our body. So I have nothing to disclose about this in terms of conflicts of interest. This was funded by the National Institutes of Health. It had many collaborators and many participating institutions, including institutions in the United States, Canada, the Netherlands, and Australia. And as everybody here knows, active surveillance is being increasingly adopted by patients who tend to have lower risk prostate cancer to avoid the side effects of treatment. Patients are very often more concerned about the side effects of the treatment than the tumor itself. And a substantial proportion of patients who adopt active surveillance will switch to active treatment when they find out through their surveillance that their cancer was more aggressive than they appreciated at the time they were diagnosed. So we undertook this study to investigate the clinical and germline genetic factors that were associated with patients who switched from active surveillance to go on to active treatment. And so in this study, the primary readout was the time that they were on active surveillance. So the clinical features first, this was a paper that was published by us in the Journal of Urology, and your female urology resident was the first author on this paper. And whoops. So the bottom line of the clinical findings, as no one will be surprised with, is that we studied almost 7,000 men, and about a third of them converted from active surveillance to active treatment with a median follow-up of 6.7 years. And the earlier conversion occurred in patients who had higher risk tumor features. And an interesting exploratory analysis revealed that in patients who had the lowest Gleason grade group 1, if they had more than 4 or more biopsy cores, they behave more like patients with higher grade tumors, so that the amount of cancer present in the initial diagnostic biopsy is an important parameter for predicting conversion. So these are called Kaplan-Meier plots. And on the vertical axis is the percentage of patients who remain on active surveillance, and on the horizontal axis is time. And so in the upper panel on the left, it shows that the patients who had higher Gleason grade tumors came off active surveillance quicker. The panel to the right shows patients with higher PSA came off active surveillance more quickly. The two panels on the lower left show patients who had a higher tumor stage or a greater number of biopsy cores came off active surveillance quickly. And the panel to the far right is risk classification that includes all three of these parameters: Gleason grade, PSA, tumor stage, or number of positive cores. Separates very dramatically those who remain on active surveillance and those who come off. Now, this just shows the hazard ratio for coming off. And so we looked at patients who had low risk, intermediate risk, high volume, low risk, and high volume, high risk. And we looked at the hazard ratio for them from converting from active surveillance to treatment. And we arbitrarily assigned the patients who had low volume, low risk, a hazard ratio of 1.0. And so you can see the patients who had intermediate risk disease had a hazard ratio of 1.78. But the patients with high volume, low risk disease, the hazard ratio was 3.46, which was nearly as high as the patients who had high volume, high risk disease. So that was an interesting finding. And then subsequently, this is a paper that came from the Weill Cornell group showing that tumor size, or the amount of cancer in the biopsy specimens, is an independent predictor of aggressive prostate cancer using multiple aggressiveness parameters independent of Gleason grade groups. So again, this confirms that even if you have the lowest Gleason grade group, if you have a high number of cores, you can run these predictive assays, and it correlates with tumor aggressiveness. However, we found no significant difference in terms of coming off active surveillance by patients' race, by their genetic ancestry, or by whether or not they had a family history of prostate cancer. So moving to the germline genomic factors, this was a study that was really the genetic brains in this project were John Whitty, who's preeminent geneticist at Stanford and UCSF, and Yuli Jiang was a postdoctoral fellow in his laboratory, and she did these very sophisticated genetic analyses. So we're going to talk about SNPs, single nucleotide polymorphisms, and this shows you three individual people, purple, blue, and orange. And SNPs, or single nucleotide polymorphisms, occur about every 1,000 bases across the whole genome, so that we all have 4 to 5 million SNPs in our genome. And if you can see in these three individuals, the SNPs are almost identical for these three people, but this one SNP where one has an adenine, the other has a guanine, the other has a thymine, this is what makes us different from each other. So that the traits that each one of us express for susceptibility to disease, whether it's diabetes, height, weight, are determined by these SNPs. And the other thing I want to introduce is the concept of a polygenic risk score. So you can have a disease like prostate cancer, in which they have identified 451 SNPs, 451 out of 4 to 5 million, that are known to affect a man's risk for developing prostate cancer. And you can form a polygenic risk score by determining the number of SNPs that any individual has by the weight of each single SNP. So some of us might have inherited 451 of those SNPs from our father and mother together, and we're going to get prostate cancer. And some may have been lucky, and of the 4 to 5 million SNPs that he inherited from his parents, he didn't get any of these 451, and he's not going to get prostate cancer as far as we can tell. And so a polygenic risk score is basically a number where you add up the number of risk SNPs they've inherited for a trait, and then each SNP has an effect size, a weight size. And so this is really just the sum of the number of SNPs multiplied by the dosage of each SNP. So these are the 451 SNPs, and for a SNP to be validated in a study, it's called a genome-wide association study. And because there are so many SNPs and so many statistical comparisons to make, for a SNP to be considered valid, the p-value has to be 10 to the minus 8. So it's has to have a very, very high levels of significance. Now, these 451 SNPs I have on this slide, they've been validated in 500,000 men. It's now 900,000 as of last week. And so almost 1 million men. And they, and about 40% of men who develop aggressive prostate cancer are in the top 20 percentile of the SNPs. And in the bottom 10 percentile of the SNPs, only about 7% of the aggressive cases end up there. So, so this is a polygenic risk score because we're talking about 451 SNPs. Now there's a monogenic SNP risk score. And sort of the classic one is BRCA2, more common in Ashkenazi Jewish people, predisposes to breast, ovarian, and pancreatic cancer in women, prostate, male breast cancer, and pancreatic cancer, and melanoma in men, melanoma in both traits. And it's a single genetic variant, okay? And BRCA2 carries a very high risk. So each of these 451 SNPs carries only a small risk by itself. But the monogenic ones are very potent. They contain a very high risk. So if you look at this curve and the black line in the middle, these are BRCA2 carriers. And this is their absolute risk of developing prostate cancer as a function of age. So if somebody is a BRCA2 carrier, by age 85, he has a 60% chance of developing prostate cancer. And the polygenic risk score sort of modulates that. So if he is a BRCA2 carrier and has a high polygenic risk score, he has an 87% chance of developing prostate cancer. But if he has a low polygenic risk score, it's 34%. So that any individual's risk of developing prostate cancer is influenced by his polygenic risk score and any mutations or monogenic risk scores. So you can have a BRCA carrier is going to have a much higher risk for prostate cancer than a non-carrier. A non-carrier, a non-BRCA carrier can have a risk almost as much as a BRCA carrier if he has a high polygenic risk score. And a BRCA carrier who also has a high polygenic risk score has an extraordinarily high risk, not only of developing prostate cancer, but of the most aggressive types of prostate cancer. So this is a polygenic risk score of men of European ancestry in the top panel, and of African ancestry in the bottom panel. And what you're looking at on the vertical axis is the odds ratio of having prostate cancer. And what you're looking at the horizontal axis is the percentile of the polygenic risk score that you're in. And so you can see as you go along from the lowest polygenic risk score percentile to the highest ones, your risk of cancer increases. Now, this slide shows a similar thing, except on the horizontal axis, we are looking at the percentage of patients who have cancer, and the cancer is divided into aggressive cancer and non-aggressive cancer by colors. So that the light green are the aggressive cases, and the light blue and African ancestry men are the aggressive cases. And just by looking at that, if you look at the percentage of the light green cases, you can see that most of the aggressive cases are in the top polygenic risk score, the 80th to 90th and the 90th to 100th percentile. And if you look in the bottom percentile, the 0 to 10th percentile, there are very few cancers, but also very few of the aggressive cancers. And this is very important because it's potentially a marker that is not used today in clinical practice that could be used in patients who are trying to decide whether they go on active surveillance. This is not commercially available at this time. So we undertook a genome-wide association study to see whether we could identify any SNPs that were specifically associated with conversion from active surveillance to active treatment. And so we carried this out in almost 7,000 men. And what you see in the top panel, what you're seeing is what we call a Manhattan plot. We call it a Manhattan plot because it looks like the skyline of Manhattan. And so the taller skyscrapers are measuring the degree of statistical significance. And across the horizontal axis, you're looking at the genome, chromosome 1, 2, 3, 22, and X. And so as we went across the genome, in these 7,000 men, we found 18 SNPs that were highly associated with failure of active surveillance, and with the p-value of 10 to the -8. And this is our discovery population. The bottom panel shows what we call our replication cohort plus the discovery cohort. And when we put those together and did the genetic analysis, several of those 18 SNPs dropped out, either because they were extremely rare. To be a SNP, you have to be present in at least 1% of the population. If you have a variant that is more rare than that, then it's not considered a SNP. So if they were extremely rare, they dropped out, or if they didn't replicate in the discovery or replication cohorts. So we ended up with about 8 SNPs. Well, you can't construct a polygenic risk score from 8 SNPs. I mean, you have to have at least several hundred SNPs. So we were kind of stuck there. We had discovered 8 highly significant SNPs that that were associated with failure of active surveillance, but they weren't enough SNPs to construct a polygenic risk score. Now, maybe if we had 100,000 patients, we could have identified more SNPs and could have done a polygenic risk score. So what we did was we looked at the polygenic risk score for prostate cancer risk. This is just simply prostate cancer risk. And we wanted to see whether that polygenic risk score would associate with failure of active surveillance. And this shows it does. You can see that as you go from left to right, it goes up like the polygenic risk score, so that the patients who fall in the high polygenic risk score for prostate cancer susceptibility also fall in the higher risk category for prostate cancer or for active surveillance failure. And this was just a couple weeks ago. This was validated by this group at University of Southern California, Chris Heyman's group, who's probably the preeminent prostate cancer geneticist, in which they looked in using the same polygenic risk score in African American men, and they found that they could predict aggressiveness of the prostate cancer using the polygenic risk score. And they didn't define aggressiveness only as failure of active surveillance, but they defined it as having high-grade disease, having advanced disease, having metastatic disease, or having fatal prostate cancer. Then there is a paper that is under review by one of the large genetic journals from the Million Veterans Study, in which, using the latest and greatest 451 genome or SNP polygenic risk score, that shows that in Black men, white men, and Hispanic men, the polygenic risk score predicts aggressive prostate cancer. So that it appears that we're getting to the point now where polygenic risk scores can be used to predict aggressiveness of prostate cancer in men who are considering active surveillance. And then we did something else. PSA levels in men are also genetically determined. So that just like some men are short and some men are tall, some men are low PSA producers and some men are high PSA producers. And the slide must have timing on it. And so we developed a 128 SNP polygenic risk score for PSA levels. So in other words, you can take a man, you can measure his, you run a chip on him, and then you can see whether his PSA levels are artificially high, artificially low, or artificially normal. And so we tested to see whether the polygenic risk score for PSA would also correlate with failure of active surveillance. And you can see that the trend is just the opposite. That the men, as you go from low polygenic risk score to high polygenic risk score, the failure of active surveillance was less. And this makes sense because men who have a high polygenic risk score for PSA have high PSA levels throughout life. They're more likely to be biopsied early in life when their cancer is in a less aggressive earlier stage. And men who have genetically low PSA levels are more likely to undergo biopsy when they're older, be diagnosed with prostate cancer in a more advanced stage, and have more aggressive disease and a greater risk of having aggressive disease and failing active surveillance. So concluding from these studies that both clinical and pathologic factors, and for the clinical factors, notably the number of low-grade prostate cancers in the biopsy specimens, predict the likelihood of conversion from active surveillance to active treatment. And these are parameters we all use in clinical practice today, except many of us don't use the number of positive cores of Gleason grade group 1, but I think that we should. And the other thing is that these germline genetic factors can be put together commercially, I believe, including a polygenic risk score created from the prostate cancer risk SNPs and an independent polygenic risk score created from the PSA SNPs in combination with measuring monogenic risk scores like BRCA2, HOXB13, and other high-risk prostate cancers. These can be put together in a genetic panel that could help that could help patients who are trying to decide whether they want to undergo prostate cancer or not. These help them make that decision, and if they do decide to undergo active surveillance, to determine what the appropriate intensiveness of their surveillance regimen should be. If they are very high risk, they might need to have a very intensive surveillance regimen. If they have a very low risk of failing active surveillance, maybe they don't need to be biopsied as often or surveilled as intensively. So in terms of my acknowledgements, I just wanted to comment on the Woody Lab that did the heavy lifting for the genetic work and my research group at Northwestern. And then I wanted to just say one final thing. For the past 3 or 4 years, I have been working with the NCI to try to develop a program project grant. The goals of it are to improve the uptake of active surveillance, because it's 80% in Sweden and it's 50% in the United States, to improve the quality of active surveillance and to do it at a lower cost. And so we're working on a grant now. It's going to be submitted in September, and we have 3 projects. One project is called Implementation Science, and this is to try to get physicians to do the right thing. And in some practices, you have patients who will put 0% of their low-risk patients on active surveillance. And you have other patients who will put 100% on active surveillance, and you have everything in between. And so if you look at the Michigan group, the music group, there's tremendous variation across the United States. Whether you do active surveillance it may be determined more by the doctor you go to than by the disease and the genetic factors that you have. So we want to improve the uptake and the quality of active surveillance. Implementation Science Project 1 and your friend and mine, Michael, is really a player in that project. He's going to be one of the leaders in the project. The second part is to develop better genomic markers, better imaging markers. And there's a lot of variability in pathology as well, and to use artificial intelligence to get rid of some of the pathology variation. And then the third project is cost effectiveness. We would like to deliver better care at a lower cost. So thank you.
Questions?
So, the patients make the decision. So if I have a patient who has 4 positive cores or 5 or 6 positive cores, I tell them that they're at a high risk to fail active surveillance. There's a very high likelihood that within a few years, they're going to have a radical prostatectomy or radiation therapy. But when I follow them, one of my colleagues told me, who's a urologist, that her ophthalmologist called her up and said, "Hey, you haven't had your eyes examined this week." And she's been busy and everything else, and she forgot to have her eyes checked. That's how you follow them. They're high risk, and you call them up and you say, you have a high rate, you're a high risk active surveillance patient, and you need to come in for your MRI or your biopsy or your PSA or your rectal exam. So I do, but I really encourage them to have more intense surveillance. Yes. We did not look at PIRAD score because most of these patients in our trial of the 7,000 patients did not have MRIs. Yes.
Might be interesting to look at those patients with prostate cancer. If we look at the SNP analysis correlated with their testosterone level, you get a similar curve as you do with the PSA.
And I'm sure it would. I'm sure it would, because testosterone secretion is also genetically determined. Yes.
High volume disease when it comes to saturation biopsies, like PIRAD-4 area, opposed to take that into account. The second question I have is prostate cancer detected by TURP different than a prostate cancer that's detected by
Okay, so the answer to your first question is that it's a very valid point that you raise. If somebody has a PIRADS 5 lesion and you take 9 cores through that lesion, you're going to have, you're going to have non-positive cores. So I think that we need to standardize how many cores you take through any PIRADS lesion, and we take 3. We try not to take more than 3. And so that at least gives you standardization. And then, what was your other question? Yeah. So if you look at the somatic genetics of prostate cancer, the transition zone, the central zone of the prostate is genetically less aggressive. So that probably a low volume prostate cancer detected by TURP is probably less aggressive than one that would be detected by a targeted biopsy especially with a PIRADS lesion in the peripheral zone.
There's one last question. Yeah.
Just correlation with volume, basic volume.
Correlation with the volume of the prostate. Well, if a man has a big prostate, you sort of allow him to have a higher PSA, right? But in terms of biopsy cores, I don't think it's the same. So if a man has a very big prostate and if he has 5 or 6 positive cores, especially if they're all not in the same area of a PIRADS lesion, then I think that the concern there is that higher risk for failing active surveillance.
Okay, so we'll move on to the last speaker of today's event. Again, Dr. Patrick Walsh from Johns Hopkins. Dr. Walsh was planning on joining us in person this morning. Unfortunately, he did have a personal emergency, so he could not be here in person. Nevertheless, he was gracious enough. This was an important event for him, so he wanted to give a presentation or still proceed with the talk that he was planning on giving it to us. So again, Dr. Walsh graduated from Case Western Reserve University Medical School in 1964. Then he finished his residency training at UCLA in 1971. And then ever since then, he has been at Johns Hopkins, and he's been there as a professor and chair until when he's retired a couple of years ago. Dr. Walsh will be giving his talk on the discovery or his experience with the discovery of a nerve-sparing radical prostatectomy. Dr. Walsh? Can you hear me? Can you hear me? Okay, go ahead.
Hey, can you hear me? Well, thank you very much, Isaac. Again, I'd like to begin by saying how sorry we are not that we are not there, especially to Dawn. Peg was really looking forward to it, but she fell and had surgery, and I'm actually calling you from the hospital. So Bernie, you were my role model, maybe the better word is hero, and my friend. I entered urology, my urology residency in 1967, and that's when you became the chair at Yale. And during my residency and the early years of my time at Hopkins, I admired what you did so much. You were charismatic, you were a fantastic speaker, you had a broad knowledge of multiple areas of urology and urologic surgery, and I learned a lot from you. And then in 1979, we started a friendship. We both elected to the Clinical Society of Genitourinary Surgeons, which is a group of 25 urologists under the age of 55 who travel once a year to one another's institutions. And the nice thing about it is you're shuttled in buses many places. And so I began sitting with Bernie on those buses, and I learned so much from him. And I talked about what I was doing. He talked about what he was doing. Over time, our wives began to attend, and my wife Peg got to know Norma very well, and Dawn, and it was an exciting time. Beginning in 1979 to 2019, a 40-year friendship. So Bernie, this talk is for you. So when Isaac invited me, he said, what would you like to talk about? And I said, well, a couple of weeks ago, I was in Milan and speaking at the European Association of Urology meeting. They're celebrating their 50th anniversary, and they had a one-day symposium on the history of discoveries in urology over the last 50 years. And they invited me to speak and gave me the title, Nerve-Sparing Radical Prostatectomy, a European Discovery. And the answer is yes. I'll now tell you the story. I have no conflict of interest to report. Historical perspectives. In 1904, Hugh Hampton Young at Hopkins performed the first successful radical perineal prostatectomy for the cure of prostate cancer. In 1947, Terence Millen, an Irish urologist working in London, described radical retropubic prostatectomy. But by the 1970s, radical prostatectomies were rarely performed despite adequate cancer control because of side effects. Major bleeding when done retropubically, virtually every man was impotent, and sadly 10 to 25% had total incontinence. And with the development of external beam radiation, surgery fell into disuse. So in 1974, when I arrived at Hopkins as the third director of the Brady Institute, radical prostatectomy was rarely performed, even at the center where it had been pioneered 70 years earlier. Now, during the prior decade, I trained at outstanding centers on the East and West Coast. And during that time, I never heard anyone suggest that these complications might be preventable. They looked on them as the price patients had to pay for the possibility of cure. And at this time, I wondered why they occurred and if it was possible to prevent them. And because I was a successor of Hugh Hampton Young, I felt the personal responsibility to solve the problem. I decided to first look at what was known about the anatomy of the structures around the prostate. Reduction in side effects through studies of the periprostatic anatomy. Why was there bleeding? Because the anatomy of the dorsal vein complex and Santorini's plexus had not been charted. Why was there impotence? Although the location of the cavernous nerves responsible for erection was not known, because all men were impotent, it was universally assumed that these nerves ran through the prostate and preservation of potency would be impossible. And why was there incontinence? Because the location of the sphincter responsible for passive urinary control was not known. So why were we so ignorant? Because the study of the periprostatic anatomy in cadavers was difficult, because after death, the abdominal viscera compress the pelvic organs into a thick pancake of tissue, and formalin obliterates the soft tissue planes. And what was the solution? Using the operating room as an anatomy laboratory and fetal dissections. So for those of you who would like more details, this article was published on the 25th anniversary of the first nerve-sparing radical prostatectomy in the Journal of Urology in April 2007. Anatomical observations, bleeding. Because of frightening bleeding, surgeons did not want to operate. So I looked at this first. Now we know the venous complex is over the anterior surface of the prostate, but it is beneath dense fascia. And because the anatomy was obscure, surgeons believed that excessive bleeding was inevitable. However, using the operating room as an anatomy laboratory, I identified a common trunk over the urethra. And this important observation led to a surgical technique that reduced blood loss. And this bloodless field made it possible to perform a safer, more precise, and thorough cancer operation. Anatomical observations on impotence. Well, soon after developing the technique for controlling bleeding, a 58-year-old man returned 3 months following surgery and told me that he was fully potent. I mean, how could that be? If the cavernous nerves ran through the prostate, this could not happen. But from this one case, I knew that they did not run through the prostate. But where were they? The answer, which was not in any anatomy book, was found on a trip to Lytton. Here is a photograph of Professor Peter Donker, a neurourologist who was the professor and chairman at the University of Leiden. In 1977, while attending a meeting, my wife and I went to a restaurant downtown. As we walked to our table, I noticed an older man who looked lonely, standing by himself, and impetuously went up to him and asked him if he was alone, why he was in town, and if he would like to join us for dinner. His name was Peter Donker. He was attending the same meeting, and like us, had been advised by the concierge to go to the same restaurant. Well, after a wonderful dinner and conversation, I thought that was the end of it. No. 4 years later, 4,000 miles away, we met for the second time in Leiden in February 1981. Donker was retired, and I was invited by his successor, Udo Jonas, to spend 5 days as a visiting professor at the Boerhaave Surgical Symposium, operating, lecturing, visiting laboratories. Finally, on my last day, Friday afternoon, February 13th, 1981, Peter wanted to repay my kindness and volunteered to take me to see a windmill museum. When I asked what he was doing in retirement, he said he was working in an anatomy laboratory. And with no premonition of what I was going to see, I said I wanted to go there. Now, in his laboratory, he was using a dissecting microscope to study the innervation of the bladder in a stillborn male infant. When I asked to see the branches to the corpora cavernosa, he said he had never looked. 3 hours later, we identified them outside the prostate. So here is my primitive drawing from that day, and there they were. And here is our formal drawing that we published, and you can see where the cavernous nerves are located and the pelvic plexus. Well, that day we agreed to continue working on the problem separately. 1 year later, in March 1982, we met again. He had confirmed our original anatomical observations with repeat dissections and with microscopic step section reconstructions. I presented my findings. Based on our original observations, we knew where the cavernous nerves were located in a tiny fetus. But how could we identify these microscopic structures in the adult male pelvis? It was like trying to find a defective transistor in your television set. So when I returned to Hopkins in 1981, I again used the operating room as an anatomy laboratory. And while performing a radical prostatectomy in October 1981, I identified some vessels that ran in the same direction as the nerves in the fetus. And based on this observation, I speculated that this neurovascular bundle could be used as the intraoperative landmark to identify the microscopic nerves. Merrill Saltzman, who was a radiologist many years ago at the Brigham, said, "You only see what you look for and recognize only what you know." So Donker agreed with this idea. Well, in March 1982, I returned home and performed a radical cystectomy on a 60-year-old man using this technique. I'd never heard of a patient who was potent after a cystectomy, but on his 10th postoperative day, he awoke with an erection. I now knew that we were on the right track. And 1 month later, on April 26, 1982, I performed the first purposeful nerve-sparing radical prostatectomy. And here's a photograph of that patient on the 25th anniversary of that operation. The patient was fully functional within a year of his operation and died 35 years following surgery, free of cancer. Well, to confirm our concept of the neurovascular bundle, Herb Lepore and I took an adult cadaver that was perfused with Bouin solution shortly after death, removed all the pelvic organs en bloc, performed 10,000 whole mount step sections with 3D reconstruction, and documented the constant association of the vessels with innervation to the corpora cavernosa. And based on those observations, Peter Schlegel performed this beautiful fresh cadaveric dissection, which was illustrated by Leon Schlossberg. So this is the picture I have in my mind of the pelvic plexus resting on the lateral surface of the rectum. Note its association with the seminal vesicle here, which is green, and then as the neurovascular bundles travel to the corpora cavernosum. The final piece of the puzzle, improving cancer control, the fascia surrounding the prostate. At that time, the only fascia surgeons understood was Stenon-Viet's fascia. But based on whole mouth step sections, the importance of the lateral pelvic fascia became evident. And this article, which was published 40 years ago this year, put everything together. The neurovascular bundle is outside the prostate, between the levator and prostatic fascia. If nerve sparing is performed correctly, the prostatic fascia, which is the same as the prostatic capsule, they are not distinct structures, must remain on the prostate. Now let me show you what we did with the old technique. The prostate was bluntly released from the rectum with the index finger, tearing it from the neurovascular bundle, which was adherent to the rectum, leaving no soft tissue on the prostate. The neurovascular bundle was then cut during ligation of the pedicle and left in place. So this is the plane of dissection in a nerve-sparing radical prostatectomy. I'm next going to show you the plane of dissection with wide excision of the neurovascular bundle. This removed more tissue than was ever possible before, making it a better cancer operation. So here you are outside the levator fascia completely, and you can see the amount of additional tissue that was never removed before. Finally, in this article, Joseph Eggleston, the director of surgical pathology, looked at the surgical margins removed by perineal, standard retropubic, and nerve-sparing techniques and showed no compromise in adequacy. Impact. In 1983, only 7% of men with prostate cancer in the United States underwent surgery, and radiotherapy was too underpowered to cure. Essentially, no one was being treated with curative intent. However, by 1993, 70% of men in their 50s and 55% of men in their 60s underwent surgery, 100,000 men that year. And over the next decade, with the availability of a safer surgical procedure that had fewer side effects and PSA screening to identify many more men with curable disease, deaths from prostate cancer declined 33%, greater than for any other cancer in men or women. At this time, when radiation therapy was still too underpowered to cure, what would have happened without Peter Donker's contribution? So at this point, I'd like to stop and honor Leon Schlossberg, who was a gifted artist whose anatomical and surgical drawings taught urologists around the world. Well, this story would not have happened if a patient had told me he was potent following surgery and I hadn't listened and wondered why. And I had not invited an older, lonely man who was a total stranger to dinner. And I had not met him 4 years later, 4,000 miles away. And I'd gone to the Windmill Museum and not asked to see what he was doing in his retirement. Well, how did this happen? Well, if you ask Yogi Berra, he would say it was too much of a coincidence to be coincidental. And if you asked Albert Einstein, he would say coincidence is God's way of remaining anonymous. And if you ask St. John Paul II, he would say that in divine providence, nothing is a coincidence. And this is what I believe. This image hangs in my office. I believe there is a divine creator, that we physicians have the sacred responsibility to care for his children, that whenever we face difficulties, we should feel confident that he will always be there to help us. And I have asked him on many occasions for that help. When I couldn't find the location of the cavernous nerves outside the prostate, I asked the Lord to open my eyes. And around that time, I met a lonely man in a restaurant. And for reasons I cannot explain, I invited him to dinner. And that is why I was in Leiden visiting Peter Docker on the afternoon of February 13th, 1981. And why I have the honor of speaking to you today. One final story, my Uncle Harry. The backstory behind the neurovascular bundles. So let me tell you about Uncle Harry first. My Uncle Harry went to Carnegie. I lived in Akron, Ohio, and my Uncle Harry went to Carnegie Tech for a couple of years, but then came back to Akron and had to run his father's plumbing business. Uncle Harry would much rather be out in nature than looking underneath a sink. And so when I was about 8 years old, he saw that I had the same interest, and we began taking hikes on Sundays, catching turtles and snakes, hunting for fossils and arrowheads, and one summer going to Canada with a Geiger counter to hunt for uranium. And he loved to teach me big words like superheterodyne. But one day he taught me the auricular ventricular bundle of His. Now I know you're sitting there scratching your head. It's why did he teach that? I just do not know. Maybe it was the alliteration that he thought would be interesting. So after these hikes, we would go to his home where Aunt Anna, my mother's older sister, a nurse, would make dinner. Gave me a chance to talk about medicine. Well, that night I looked in her nurse's dictionary and learned that the neurovascular bundle, that this was the neurovascular bundle between the auricle and the ventricle. So from the time I was 8 or 9, I knew that autonomic nerves traveled in association with adjacent structures. For this reason, when I saw the capsular and arteries of the vein, I instantly knew that they could be the landmark I was looking for. You only see what you look for and recognize only what you know. Now, at that time, the word neurovascular bundle, I looked up recently in the 1983 Gray's Anatomy, there was one reference in the index, and that was not in the genitourinary section, nor was it on the page that they referenced. During the final years of my residency, Uncle Harry developed metastatic prostate cancer and died a painful death with Aunt Anna at his side. So today, if you go to Stedman's Medical Dictionary, the 27th edition, published in the year 2000, and you look under the word bundle, in the left column, you will find the atrioventricular bundle of His. And next to it, in the right column, you will find the neurovascular bundle of Walsh. Thank you, Uncle Harry, and thank you, Isaac, for inviting me to give this talk.
Thank you, Dr. Walsh, for the inspiration, the great talk. Can you take a couple questions, Dr. Walsh?
Sure.
Okay. Any questions for Dr. Walsh from the audience? No? I think that, okay. Dr. Walsh, I think the talk was just too inspirational. I think it was, we were just awestruck. So there's no question from the audience here. So again, thank you so much for taking the time on a Saturday morning to join us.
Well, thank you for the honor of doing it.
Okay. All right, so we are here to the end now, last. So again, I want to thank every one of you for being here today. And again, just being here together and remembering what Dr. Lytton has meant to each one of us. I think it's just been a great event. And just again, remind you that again, as Yale Urology moves forward, there are multiple events looking to the future where we would love to continue to have these gatherings and just continue to engage each one of you. So we have the alumni reception coming up in AUA in Chicago. We also have our visiting professor day in June with Dr. Ash Tewari visiting us this year. And then finally, again, just to the inaugural event that we're having, this is again another event that we're going to be putting as a part of our, as cementing our legacy in urology, that we're going to be launching the Yale Urology Inaugural Symposium. We call the so-called the Bulldog Debates, the name to be changed maybe. Again, this is going to be held at the Yale Club in New York City on date September 30th to October 1st. So would love to have the alumni engaged on this event. So again, I am really grateful for each one of you on behalf of Dr. Lytton family, Department of Urology at Yale. Enjoy the rest of the day. And there's going to be, I think, small reception later on for us to get together again and continue our conversations and continue our relationship. So again, thank you and have a safe trip back home.